A very good morning to all. I, Dr. Radha Chauhan, welcome you all on the behalf of all the organizers, Sanjay Gandhi Postgraduate Institute of Medical Sciences, Guru Angad Dev Veterinary and Animal Sciences University, Indian Virological Society, American Society for Microbiologists. Before we start the proceedings, let me first acknowledge the presence of some of our eminent guests. I would like to welcome our Honorable Director, Sir, Professor R.K. Dhiman, Sir. I would like to welcome our Honorable CMS, Sir, Dr. Gaurav Agarwal, Sir. I would like to welcome our Honorable Director, IDSP, Dr. Vikasendu Agarwal, Sir. I would like to welcome our Honorable Secretary of Indian Virological Society, Professor Shailendra Saxena, Sir. I would like to welcome our Honorable Organizing Secretaries, Professor and Head, Dr. Ujjwala Ghoshal, Ma'am, and Dr. Yashpal Malik, Sir. Dr. Yashpal Malik, Sir, is the Organizing Secretary and Secretary of Indian Biological Society and Dean of Gadwasu University. Now, I would like to invite Dr. Ujjwala Ghoshal, Ma'am, Organizing Secretary, Professor and Head of Department of Microbiology, Ma'am, to speak a few words about today's CME.
Thank you very much, Radha. I assure you I will speak really a few words only. So welcome you all. I would like to take this opportunity to welcome you all this CME on COVID-19. So a very good morning and good evening also. Good evening because we have participants and eminent speakers from different parts of world, including USA, different state, Pennsylvania, Arizona, Nevada, and several others, and also from France, West Indies, and others. So time zone is different. So welcome you all. Many of you will be wondering that now this pandemic is largely controlled, cases are decreasing. So what is the need of this CME at this point of time? So we think that the history is very important. And when this pandemic started or the pandemic came, we did not know anything about the virus or the disease or treatment or anything. But in past 2 years, we have lost a lot, but on the other hand, gained a lot. As each coin has two sides, so we have learned a lot. So we thought it is very important to discuss all this. And also we saw that whenever we are a bit relaxed, that now everything is over, we are very happy, then we make some mistakes and the next pandemic comes. So we are also apprehensive that what is the future, whether we'll again face another wave or the so-called fourth wave. And in fact, this fourth wave has already come in neighboring countries like China, Malaysia, and few others also. So we thought that it is very important that we discuss what we learned, what is ahead. And I'm very thankful to American Society of Microbiology for electing me as their country ambassador for the Northern India. And also to Dr. Yashpal Malik, who joined hands with us and helped in organizing this CME and helped us every stage. And also Indian Virology Society, Guru Angad Dev University, Gadwasu University, where he works. And also I'm also very thankful to all the speakers and the delegates who have showed, though we started a few days back, but the enthusiasm is so much that we have more than 1,000 registered delegates, not only from various parts of India, but various parts of the world, as I told you. So with this world, I once again welcome you all and thank you all for your participation. Thank you.
Thank you, ma'am. Now I would like to invite Secretary, Indian Virological Society, Professor Shailendra Saxena. Okay, sir. Now I would invite Director, Sir, Professor R.K.
I'll go there.
नहीं बैठना
First of all, thank you, Dr. Ujjwala Ghoshal, for the invitation to this CME program on COVID-19. And congratulate you and your team also for organizing this activity in association with the American Society of the Microbiology. Dr. Gaurav Agrawal, CMS, Dr. Ujjwala Ghoshal, HOD Microbiology, Dr. Uday Ghoshal, HOD Gastroenterology, my dear faculty, students and delegates, and the speakers who are part of this CME program. First of all, I would like to tell you, whenever I talk about the COVID-19, it reminds me my joining this institute in the February 2020. Because just after that, the first wave of the COVID-19 pandemic arrived in Lucknow or in the state in the month of the March 2020. And thereafter, what we have seen so far in the last 2 years, there are now a total of the 3 waves. And almost every wave is starting either in the March or in the April also. It gives us a lot of learning also. But it reminds me that how we tackle it, it gives me the importance of the team approach as far as this COVID-19 epidemic is concerned. Ek minute ruk jao. Just let me concentrate also. And the first epidemic, out of these 3 epidemics, the second epidemic was the severest one. We all know the variants, different kind of the variants they are causing these epidemics. And the first epidemic, it was caused by the alpha and the beta. And the most severe one that arrived in 2021 was by the delta. And the latest, which was not that severe, Omicron. As far as any containment of the infection is concerned, there are many factors. Not only it is the virulence of the virus, but also on the defensive side, the vaccination that we have done in our population also, or the exposure to the symptomatic or asymptomatic infection, ultimately leading to the immunity at the individual level as well as the herd level. So that's what I am trying to tell you, that over the 2 years, what we have seen, not only the control of these different waves that we have seen with our team efforts at the national as well as the state level. And I take this opportunity to laud the efforts of the center, especially our Honorable Prime Minister, that we have achieved the target, not only the target of the vaccination in whole population across India or across the nation also. I am aware now more than 190 crores vaccination doses has been given. And recently also that they are covering the children also, 12 years of the age plus also. So looking at the interaction and the variants also, you all know what is the variant of the concern. If it is more transmissible or it is causing the more severe disease and it is evading the vaccine response also. So these kind of the variants that we have observed, alpha, beta, delta, and latest that is the Omicron also. The team effort that we have given, not only there were several challenges in front of us, particularly in the second wave, when we have lots and lots of the patient, it was difficult to admit because the system was overwhelming at that time. But nevertheless, I again advocate as far as the state policy was concerned, a number of the beds were created. I would say more than 2.5 lakhs or the 2 lakhs of the beds for the admission for these patients was created, and all kind of the drugs that was required, including oxygen therapy and other drugs, they were available for the free of the cost. So the challenges were met also, and during the same time we have seen the change in the behavior of the virus. You all know these are the RNA viruses, and whenever they replicate and they have the variations or they have the mutations in the genome also, particularly in the spike protein also. But over the time, what we have seen, the virulence of the virus after the Delta virus has gone down. And we are listening to that other variants are coming. But to my knowledge, whatever the new variants of the Omicron is coming, that is not more lethal and not more transmissible also, and it is not more evading to the vaccine response also. So what is the situation as of now? As of now, if we see the interaction between the immunity and the virus virulence has gone down dramatically as a result of the new variants, which are not more virulent and not more transmissible also. And but on the other hand, now we have the vaccination rate, which is causing the herd, that is contributing to herd immunity. Why I'm saying the contributing? Because the herd immunity comes not only from the vaccination, but also from the exposure to the virus or exposure to getting the infection also. So at the end, I would like to end the message over here that we are now, as of now, in a very comfortable position as far as the new variant of the virus is concerned regarding its virulence, transmissibility, as well as the evading response to the vaccination, and the immunity that we have, herd immunity that we have. So I personally feel that this is the beginning of the end of the epidemic or the pandemic, and what we are going to have new variant may be of concern or may not be, but not more virulent in years to come also, which can be tackled without much problem also. Thank you very much. Opportunity.
Thank you, sir. Moving forward, now I would like to invite our CMS, sir, Dr. Gaurav Agarwal, sir, to say a few words.
Thank you and good morning. Professor R.K. Dhiman, the Director, SGPGI, Dr. Vikasendu Agrawal, Director, IDSP, Dr. Yashpal Malik from the Guru Angad Dev Veterinary and Animal Sciences University, Professor Ujjwala Ghoshal, Head Microbiology, SGPGI, all my faculty colleagues here, the various speakers and delegates who are connected from various parts of the world, all my younger colleagues from SGPGI. Firstly, I would like to thank the organizers for the opportunity for me to participate in this function. The CME on SARS-CoV-2 or COVID-19 is very relevant, very pertinent to our understanding of this pandemic. And if I may say, this pandemic, which is transitioning into an epidemic with all the efforts put in by scientists from across the globe and the administrators, as Professor Dhiman said, the various policies which are government, both at the central level and the state government level implemented, has helped us control this behemoth of pandemic. And more or less, we are at that juncture of time where we can breathe easy now, having curtailed the transmissibility, having controlled the disease with the vaccination and various other strategies which have been put place. So I think it is praiseworthy that the Department of Microbiology, in association with the American Society of Microbiology and the Indian Virological Society, has thought of this CME. And I wish all the delegates to take good lessons from whatever is discussed over the various sessions planned for today. And I wish all the success to this CME. Thank you very much.
Thank you, sir. Now I would like to invite Secretary, Indian Virological Society, Professor Shailendra, to say a few words.
Good morning, and all the dignitaries present here in the audience. Professor Dhiman, Honorable Director, SGPGIMS, Dr. Malik, Secretary and Dean at Gadwasu, Professor Ujjwala Ghoshal, the organizing secretary for this meeting, and the ASM ambassador in India, and several other renowned honorable people, Dr. Ghoshal and Dr. Agrawal. So many are, Dr. Vikasendhu I can see here, and several others who are present in the audience. And my old friend, Dr. Kuchipudi, Suresh, who is also online, Dr. Rathu, and several others. I really welcome all of you from the core of my heart and on the behalf of Indian Virological Society to this CME, which is jointly organized by the SGPGI, Gadwasu, IVS, and ASM. As you may be aware, the Indian Virological Society is a member of the International Union of Microbiological Society, IUMS, which was established in 1984 with the objective to promote the research and development in the field of virology. It is a unique scientific body that provides platform for those associated with the characterization and management of viruses affecting animal, human, fish, insect, plant, and other living organisms. The society always annually organizes various national and international conferences, trainings and seminars, workshops, invited lectures, academia, industry meet, etc. The society recognizes the outstanding contributions of the individual members by electing them as a fellow of the society and conferring them the Lifetime Achievement Awards, Springer Nature IVS Best Publication Award, Young Scientist Awards, and so on. The society has very close collaboration with the World Society of Virology, which is emerging as a leading world forum for addressing problems related to the cross-boundary viral epidemics or pandemics. IVS publishes a biennial newsletter and a very prestigious internationally reputed journal called Virus Disease in collaboration with Springer Nature. It covers both basic and applied areas of virology, and I specifically look forward to get the manuscripts to be published in this particular journal. Last year, the journal received the manuscripts from at least 32 countries around the globe, and currently, it has been cited and indexed in most of the prestigious databases, including PubMed, Scopus, Embase, and so on. And the site score is 3.4. So once again, I welcome you on the behalf of IVS and wish grand success to the CME and look forward to more collaboration with ASM, SGPGIMS, and Gadwasu, as well as IVS support is always for you. Thank you very much.
Thank you, sir. Now, I would like to invite Director IDSP, Dr. Vikasendhu Agrawal, to say a few words. Now, I would like to invite Dr. Yashpal Malik, Secretary, Indian Virological Society and Dean, Gadwasu University, to say a few words.
Am I audible to everyone?
Yes, yes.
Yes, sir, you are audible.
Thank you very much. Very greetings from Gadwasu. And good morning to one and all present over here for this CME on COVID. I'm Dr. Malik, Secretary General of Indian Virological Society, Dean Biotechnology at Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana. And at this moment, I take the opportunity to propose vote of thanks for all those who are associated with this program. And this is really a concept that we are proving in One Health, that two institutes, SGPGI and Gadwasu, both came forward under the aegis of American Society for Microbiology and Indian Virological Society. Both joined hands together to discuss some of the issues which are challenging world over, like COVID. This is one example, and then there are many more, like Japanese encephalitis, Zika, many more. We have to come forward and sit together to have some common strategies to fight and prepare for the future upcoming problems. I'm thankful to President and Secretariat of the ASM, President and Executive Council members of Indian Virological Society for permitting and giving this opportunity. Special thanks to Director of the SGPGI, Dr. R.K. Dhiman, Director Infectious Disease Surveillance Program, Dr. Vikasendhu Agrawal, Dean of the SGPGI, Dr. Anish Srivastava, CMS, Dr. Gaurav Agarwal, Faculty in charge is research, Dr. UC Ghosal, faculty and staff members of microbiology, and some of the faculty members who need special attention is Dr. Nidhi Tejan, Dr. Akshay, and Dr. Nikhil Sharma. I'm also thankful to all the faculty members, students who are attending this conference nationally and internationally. The chairpersons who are in upcoming sessions, they are taking care of some important issues. The speakers from national and international platforms who are joining, and some of them are at odd hours also, but we are thankful to all of them for sparing time and giving their consent. All the delegates. At this moment, I also like to thank my worthy Vice Chancellor, Dr. Inderjit, who is a dynamic personality in the area of research, academics, the press and media who are associated with coverage of this important issue on COVID. And in the last, I would like to thank anybody if I left by name. Thank you very much, Dr. Ujjwala, for coming up with this program. And I think we are more than 2 months we are in discussion, finalizing daily mailing to some of our international leaders in the COVID area. And ultimately we came out with a very good technical program, and hopefully everyone who is attending this program will see that some of the good way forwards will be suggested, recommendations will be there, which will be useful to the community overall. So thank you very much, each and everyone. Dr. Ghosal, please.
Thank you, sir. Now, I would like to invite faculty in charge research and Professor and Head Department of Gastroenterology, Dr. UC Ghoshal, sir.
Good morning, everybody. I thank the Department of Microbiology and the organizing team and all other supporting societies who organize this program. The program is very timely because we had the COVID pandemic. The whole world was devastated, including India. And now you, the microbiologist, had played a very key role along with the government and the policymakers and all other group of people, including media, to control this pandemic. So actually, that's why we can live now better. And we clinicians, we can function now more normally than the time when the COVID pandemic devastated. We know how non-COVID care was in jeopardy during the COVID pandemic. So it is very timely program. And actually, I am working as the faculty in charge research in SGPGI Lucknow for last 2 years. Actually, it is like Dean Research, and our aim is to bring research in the forefront in the institute. Because if you look at the 3 pillars of the institute, the one pillar is for patient care, other pillar is for education and training, and third pillar is for research. So we want, and we all know that unless there is a research in the institute, neither patient care become up to the mark, nor teaching and training become up to the mark. Therefore, research is in the forefront in our priority of the research shelf. And this kind of program actually helped to spread education, to spread knowledge on technology, bring new ideas, and motivate people to do research. So therefore, I really compliment the department and all other supporting societies for organizing this program. I also really feel very pleased to be a part of this program, because what happens is, though I am a clinical gastroenterologist, but still research is one of the priority areas of mine. And since the country was devastated, world was devastated, I wanted to little bit contribute to the COVID research, and we have worked on this field for last 2 years. So therefore, thank you for inviting me in this program. I propose, or I request, that all these societies who have come forward to support this program, there should be an output. I say, why don't you produce an Indian consensus on prevention and management of COVID-19, including all these experts, so that we get a document for the future pandemic, if ever it comes, not only of COVID or any other variant, but other diseases, so that we learn, we have a document. So I propose that you may initiate a process to do Indian consensus on management and prevention of COVID-19 pandemic. And at the end, I wish a grand success of this program. And I thank everybody in the organizing team, all those people in the Department of Microbiology who spend so much effort for many, many weeks or months to organize this program. I thank all the supporting societies for helping in organizing this program. And I thank all the participant, faculty member, and also the delegate. Thank you so much.
Thank you, sir. Thank you all for joining us today. Inaugural session is over. Now we'll start the CME after a short break of 10 minutes.
Good morning, Dr. Vettel. Good morning, Dr. Suresh.
Hi, good morning, Dr. Rama. Nice to see you.
Thank you.
It's actually pretty late here in the US, but yeah, good morning for you.
I know it must have been late night for you.
Yeah.
It's a big challenge to come for international CMEs and speak. I understand that.
But the good part is it's Friday night, so I can stay up
Oh,
a
that's
bit longer.
good. Yeah, that's good. Yeah. Hello, Dr. Ratho.
Here in the US, but yeah, good morning for you.
Good morning, ma'am. Good morning.
Good morning. It's a big challenge to. Hello, Dr. Vijaya. CMEs and speak. Yeah, so nice to see you after a long time.
Yes,
Good
ma'am.
part is
बहुत
it's
दिनों के
Friday
बाद
night,
मिल रहे
so
हैं
I can stay up
Oh, that's good.
longer.
Yeah, that's good. Yeah.
Good morning,
Hello,
ma'am.
Dr.
Good morning.
Ratho.
Good morning.
Good morning, everyone.
Here in the US, but yeah, good morning for you.
Good morning, ma'am. Good morning.
Good morning. It's a big challenge to. Hello, Dr. Vijaya.
Hello.
Yeah, nice to see you after a long time.
Yes, ma'am. Yes,
Good
ma'am.
morning.
बहुत
It's
दिनों के
Friday
बाद
night,
मिल
so
रहे हैं
I can stay up
Oh, that's
longer.
good. Yeah, that's good. Yeah.
Good morning,
Hello,
ma'am.
Dr. Ratho.
Good morning. Hello.
I think there is echo on the host and check. Big challenge to.
Looks like it's not echo, Dr. Rama. It's actually a delay in the
Oh,
audio.
Yeah.
So as
delay
we
in audio,
speak,
you're right.
we hear real time on the audio. Looks like there was some lag.
Yeah, there was
somewhere.
some delay, I think. You're right. Yeah.
Yeah.
No. No delay. अच्छा, ऐसा करके छोड़ दो। Now should I start? Good morning, everyone, and good evening also. Dr. Suresh, can you hear me?
Yes, ma'am. I can hear you nice and clear. Thank you.
Okay, thank you very much. So I think it's the time to start the session.
Good morning, everyone.
Good morning, everyone. So now we start with the keynote lecture by Professor Suresh, and the chairperson for this session are Dr. B.L. Sherwal, who needs no introduction, editor in Indian IR Journal, Dr. Chand Wattal, and Professor Rama Chaudhary. All of them don't need introduction. So I will request them all to start the session.
डॉक्टर शेरवाल. हाँ सर, आ जाइए सर. नमस्कार सर, आइए सर, आइए. ओके सर. ओके. ठीक है सर.
Thank you, Dr. Ujjwala.
I'm so sorry, please. I have to take a call.
Go ahead.
Yes,
Go ahead.
please.
No, thank you very much for joining us.
So I think we can start. Dr. Wattal, you want to say something?
Yeah, it's a pleasure to be here on this session, and I welcome Dr. Suresh, who is awake actually in the night. He's in US, and it will be a pleasure to listen to him while he's trying to stay awake. And it's a very important topic that we are trying to see over here as to what is happening across humans into the other kind of mammals and maybe even the primates, as far as the SARS-CoV is concerned. As of IJMM, we are actually looking for such properties that give us some insight. And I would even request Dr. Suresh once we are through with the CME. So this is a request which is to Dr. Suresh right away. But I would not delay this session further. So Dr. Suresh, kindly go ahead.
Yeah, thank you. And first of all, good morning, all of you. And I would like to thank the organizers, particularly Dr. Ghoshal and Dr. Malik for the invitation. And it's a pleasure to be speaking with you. And if I can bring up my slides, if you please stop sharing, then I can bring up my slides.
Okay. Stop sharing. Kar do.
Thank you. Hope you can see my slides. And may I also request all of you to kindly mute your microphone so there is no feedback. So I work at the Pennsylvania State University in the US, and I'm part of the Penn State Huck Institute of Life Sciences, which is an interdisciplinary institute that connects multiple disciplines. And for the seminar today, I would like to briefly give you an overview of coronaviruses and their zoonotic implications. I'm sure the audience know very well. And then I'll briefly talk about some of our recent research in our discovery of widespread infection of deer with the SARS coronavirus 2 in the US. And then some of the key questions, whether SARS coronavirus 2 is likely to become a multi-species zoonosis like other viruses that we know, influenza, for example, and then what are the implications to the long-term trajectory of the pandemic. So we have heard, it was mentioned in the inaugural session that the pandemic is over. And I think it is important to recognize that we are at the perhaps start of the beginning of the end of this pandemic, but the reminder is that this pandemic is not yet over. And I've just took a snapshot of COVID a map from Johns Hopkins as the inaugural session was going on. And as you can see from the global numbers, as we just 20 minutes ago, there were over 478 million cases of COVID and counting, and over 6.1 million deaths. So we are still in the pandemic, although there is the light at the tunnel that we can see the pandemic is coming to an end. And my research group, we focus on 3 specific areas of SARS coronavirus 2. And one of them is to ask the question, what is the susceptibility and the likely adaptation of SARS coronavirus 2 in animal species? And the other areas of focus is to develop the next generation COVID-19 vaccines. And we also collaborate with several engineering and other faculty to develop rapid and smart testing tools. So the presentation I'm going to talk today falls into the first area of our research, the susceptibility of animals. So just for brief background, coronaviruses are enveloped viruses. They are the largest RNA genomes, and they code multiple proteins, around 29, of which 16 are non-structural proteins. And unlike other RNA viruses like influenza, for example, that do not have a proofreading mechanism, so they make more mistakes as they replicate, coronaviruses are slightly unique in the sense that they do have a partial proofreading mechanism. So the NSP14, non-structural protein 14, acts as a exoribonuclease, which does correct mistakes made by the RNA-dependent RNA polymerase. However, the other parts of the virus are not proofread. So therefore, the virus does make mutations as they replicate. And if we look at where these viruses live and how long they've been around for, we know that the coronaviruses have been around for a long time, perhaps over 50,000 years or longer. And they're virtually adopted in most species of animals, including humans. And one of the unique features of these viruses is that unlike the negative sense RNA viruses like influenza, these viruses have a unique capability of recombination, homologous recombination. And that also sometimes contributes to emergence of novel variants of these viruses. And based on the previous epidemics and pandemics, so we know how epidemic human coronaviruses typically emerge. Viruses in their natural reservoir hosts like rodents or bats they first undergo what is called an interspecies transmission event, where the virus from the natural reservoir spills into intermediate hosts, such as domestic animals like cattle, pigs, or palm civets, or camels, and maybe pangolins and other animals. And due to the close proximity to these viruses, humans could be exposed, and that results in what is called as a zoonotic transmission event. Sometimes we call this also as spillover. And consistent with this hypothesis, when a virus that emerges from a natural reservoir through intermediate host and infects people is also likely to spill back into animal population if the conditions are right. So we have still questions about the origins of this virus. There are lots of different hypotheses being presented, but the widely agreed hypothesis is that the virus is likely to have come from a bat, although we do not know what the intermediate hosts may be, whether there are intermediate hosts or not, it's not still clear. But as the virus emerged and caused the pandemic in people, we have seen the virus infected many different animal species, both domestic as well as the zoo animals. And there were lots of cases about tigers and lions, and nearly 16 different species of wildlife in zoos have been infected with SARS coronavirus 2 exposure from humans. And there were also several reports of domestic dogs and cats being infected with COVID. And there was a classic case of mink in Denmark where several animals were culled. And then there were also experimental evidence to show if you infect experimentally, a lot of these animals are not only susceptible, but they also transmit the virus among themselves very efficiently. So we know now that SARS coronavirus 2 can infect a multiple range of animal species, including humans. And as was mentioned in the inaugural session by several dignitaries, the virus continues to evolve, which is not really surprising because any RNA virus, if it continues to infect a host, they make replication errors or mutations, and that contributes to emergence of variants. So we have seen several novel variants emerge, and more recently, Omicron variant. And one of the unique features of Omicron is that it is by far the most highly mutated variant. And one of the leading hypotheses for the origins of Omicron is perhaps the virus had evolution in an unmonitored animal species or groups of animals. Again, yet to be determined. So now switching gears to talk about a study that we have published and which was widely covered by media across the world, including the New York Times in the US, is the first discovery of natural infection of SARS coronavirus 2 into a wild animal species. In this case, it happened to be the white-tailed deer in the US. And the question is, why did we look into white-tailed deer in the first place? So computationally, based on the ACE2 receptor, lots of animal species have been predicted to be susceptible, and white-tailed deer are placed in the highly susceptible category. That is the evidence number one. And number two is that experimentally, when white-tailed deer were infected with SARS coronavirus 2 in a biosafety level 3 laboratory, these animals were not only susceptible, but they were effectively able to transmit the virus among other animals. That is evidence number two. The third evidence came from a serological survey that US Department of Agriculture has conducted. And they have found nearly 40% of deer that they found in 4 different states of the US have antibody evidence to SARS coronavirus 2 exposure. So therefore, there is sufficient evidence. And now to show that naturally in the wild settings, deer are getting infected with SARS coronavirus 2. So what we need is to actually find the viral nucleic acid in these animals. And that's exactly what we have done. So just for background, white-tailed deer are the most abundant large mammal species in North America. There are over 30 million numbers of these animals in the US. And just for context, the human population is 300 million. So almost a tenth of human population are deer in the US. And they also have a significant economic impact in the country. Various activities like hunting and recreational activities, they do contribute a lot of revenue, close to $28 billion to the US annually. And this is the map that shows the abundance of white-tailed deer density. So you can see pretty much in the most of the country, there were lots of deer around. So we partnered with one of the states called Iowa, where they have collected retropharyngeal lymph nodes from deer and they were preserved. And this is the map of Iowa. And to orientate you, these are temporally and geographically located. So we have we have information of the exact GPS location of these samples. So whenever you see a black circle, that is a negative sample, and the positive sample is denoted by a red circle. So April through August of 2020, this is the early part of the pandemic, just to remind you. And many samples we collected from different parts of the state were all negative. And in September 2020, we saw 2 positive animals that are 200 miles apart. What it indicates is that it is likely to be 2 separate spillover events and not just one location, because deer do not travel that far. And if you zoom into one of these locations, you can see lots of negative animals and 1 positive deer. And then zoom out, and then we zoom into this area here. Again, lots of negative animals and 1 positive animal. And then as the time progressed into October 2020, 1 more animal shows up. And by November 2020, there are lots of different animals are positive. And by December, many more. So it clearly indicates that after the virus naturally infected 1 or 2 deer, and there was a lot of deer-to-deer transmission. And this makes, and if you zoom out, and this is how the picture looks across the state, many different animals were positive. And we published this in the Proceedings of National Academy of Sciences. And this became the first wild animal species to be naturally infected with SARS-CoV-2 in the world. And interestingly, we were also able to map the epidemiological curves of human infections with what we found in the deer. So just to orientate you again, the left-hand side y-axis is showing the weekly cases per 100,000 human population. And the right side y-axis is the percent positivity in deer. So you see that the blue bars are the human cases. They go up significantly during November, December of 2020, where the peak of a pandemic. And nicely coinciding with that time, you also see a significant raise in the cases in the deer, which indicates that the source of infection to these animals was coming from human beings. And then we also have generated whole genome sequences from these animals. And those red bars with the deer icon, they are the lineages of SARS-CoV-2 we found. And there were different lineages, B1.234, B.1, and different ones. But the interesting factor is that these lineages of SARS-CoV-2 we found in the deer, they nicely overlapped with what was circulating in humans at that time in that part of the US. So clearly 1 more evidence to show what was in human population was also found in the deer. So it's a clear case of spillover events. And it is not only 1 spillover event, but there were independent, almost sometimes parallel, multiple spillover events were found in the deer. So, and the question is that how this can happen, because deer are wild animals and they do not come close to humans. And we know that for the transmission of the virus, you have to be less than 6 feet apart or 6 feet closer to the animals, and you don't wear a mask or things like that. And there were lots of different possibilities, although we do not exactly know how these animals may have been infected, but it could be contaminated food sources or contaminated environment, or they could be intermediary hosts. And just to remind also that a recent study found the SARS coronavirus 2 can remain alive and infectious on various types of food substances for almost 21 days. So just imagine if somebody ate, throw a half-eaten apple, and the apple has their saliva deposited. Unlike humans, when animals come in search of food, they first sniff before they eat. So they can easily pick up the infection. So that's one of the possibilities that may have happened. And after we have processed a few thousand samples, this is how the picture started to look like. And you can see a whole bunch of red dots all over the state. And this is another reminder that what would happen if we do not do any mitigation, right? So for example, if we did not do any social distancing, if we do not wear any masks, and that's what animals do. They don't social distance, they don't wear masks. There was a widespread infection that literally swept these animals like wildfire. And it is a reminder that when you have a respiratory virus that spreads, the mitigation is so critical, and especially wearing respiratory protection such as masks are so critical in preventing or at least slowing down the spread of the virus. So overall, there were nearly half of the animals were infected in just a matter of few months. So more than 60% were found to be positive. And that is story number one. And now we just shift gears and change the time, fast forward into 2022, January and February, which is just a couple of months ago, and in the middle of city of New York. And there is a place called Staten Island in the New York City, which is right in the middle of Manhattan. And we found that deer in that particular Staten Island park were also being infected, but this time a slightly different story. So if you see the map of Staten Island, all the dots in the blue are negative, and you see the red dots were positive animals. And this time these were live animals. We were able to test not only their virus positivity, but also antibody. And by genome sequencing, we actually found out of the 8 positive animals, 4 had Delta variant and 4 had Omicron variant. And this became the first report of Omicron infection of any animal species in the world. And so, which also reminds that the currently circulating variant of SARS coronavirus 2 is also capable of infecting animals, which is really important to remember. And we were also able to look at the antibody in these animals, and there was also evidence of prior exposure, as you can see by the presence of neutralizing antibodies in these animals. So again, they are geographically located. The red dots represent positive animals for antibody, and the blue dots negative animals. And they all have GPS locations, so we know exactly which part of the island they came from. And as we were completing these studies, another study came from our colleagues in the University of Pennsylvania in Philadelphia, where they found 2 highly divergent alpha lineage virus circulating in the deer. As you know, the alpha lineage was replaced by the highly transmissible Delta variant. But it appears that the alpha lineage spilled into deer, but it continued to maintain by these animals even after the virus, the variant of alpha was no longer circulating in people. So which also indicates that these animals are capable of maintaining the virus even after we control the circulation of the virus in humans, which is significant in terms of the virus ecology and future evolution. And more recently, a study came out of Canada where they have found a highly mutated SARS coronavirus 2 lineage, which they named as Ontario white-tailed deer clade. As you can see here, it is the long branch in the phylogenetic tree essentially tells you that there are lots of missing pieces that we did not monitor for. And the most interesting part is that this version of the virus was not found in humans. Therefore, it is indicative of the fact that the virus circulated in these animals and significantly mutated and potentially adapted to these animal species. And now there are 3. So obviously the question is, after the virus from humans gets into animals, and can the animals give the virus back to humans? And now we have 3 evidence to show, yes, that can happen. The first one is the story from Denmark, where initially mink in the farms were infected. Again, it was found that the humans, people who are working in the mink farms, were found to be infected with the version of the virus in the mink. That is evidence number 1. And number 2, if some of you have been following the science, recently there was a very fascinating case of Delta virus infection in Hong Kong through imported pet hamsters. So the story is Hong Kong has a very strict zero COVID policies. They're tracking every single case, and they were able to effectively control. And some of the pet shops have imported hamsters from Netherlands, and these hamsters had COVID-19 infection, Delta variant infection. So after they arrive in Hong Kong, then the virus spreads from pet animals to humans, and that created an outbreak of Delta in Hong Kong. That is evidence number 2. And the more recent one in Canada, the highly mutated white-tailed deer clade was also found to have infected one human who has contact with the deer. And that makes the third case of animal to human transmission. So now if you think about it, originally the virus is most likely came from an animal, zoonotic virus, spills into humans, and then it spills back into animal population, and then animals give it back to humans. So it is creating the full circle, if you will. So now the question is, okay, some animals randomly in some part of the world are infected. So what is the big deal? Why do we care? It is significant for a number of reasons. And this is a chart of several different animal species that could be susceptible. And the fact is that throughout the world, we are very busy dealing with public health problems, which is important because we are all focused on testing and vaccinating humans. So we have not been conducting surveillance or testing of animals. So we have largely no idea of different animals in different parts of the world, whether they are infected with SARS-CoV-2 or not. It is possible that many different wild animal species in different parts of the world may already have been infected, but we have not been monitoring, so we have no idea. So, and what could happen is that then unmonitored, the virus might be circulating in, so we know that the virus circulated in managed or captive settings, but what about the wild natural habitat? So if one animal is infected, the question is who are they giving the virus to? And then that creates multiple transmission networks, which will create or which will make the ecology and evolution of this virus significantly complicated than we currently comprehend. And similarly, if you just take one particular ecological niche, for example, in the US, the white-tailed deer also share the same space with deer mice and cats, and there could have lots of transmission networks there. So it is really important that we have to, and the reason is that if we do not address it now, it is possible that the virus could become like influenza virus. As you know, influenza viruses are extremely complicated. There are lots of different variants. It's because that they have many, many different hosts and the virus constantly circulates. And it is possible that this virus could end up like influenza if we do not pay attention. So just to summarize, SARS coronavirus 2, I showed you, infects a wide variety of animals, and there are widespread natural infection of deer that we found. And also now there are 3 cases of animal to human infections. And therefore we should consider SARS coronavirus 2 as a multi-species zoonosis, and targeted surveillance of high-risk animal species is absolutely critical. And from our own research, we are looking at many open questions, whether the virus will continue to be maintained in these animals, and some of the mechanistic studies, which I would not go into. But I also wanted to highlight what Dr. Malik has mentioned, rightly so, is that when you think about emerging and zoonotic infectious diseases, we need to think about the One Health approach because human health, animal health, and environmental health are not separate, but they are intimately connected to one another. So therefore, an integrated, comprehensive One Health approach is the more effective and efficient way of dealing with the current and the future zoonotic emerging infectious diseases. And finally, our studies had lots of media attention. So this is a picture of myself in the cover page of New York Times, and it was covered by many different entities, including BBC and the PBS NewsHour in the US. And I also wanted to mention that these studies were possible because of our biosafety level 3 lab at Penn State. And obviously, lots of people that contributed to this work, many of the members of my lab. and also the funding agencies that supported this research also are particularly important to mention here. And with regards to the DEAR studies, we have several collaborators across the world, including University of Cambridge and many different universities in the US that have contributed to this study, and the New York City Parks and White Buffalo, which is a conservation group. And that's the end of my presentation, and I'll be very happy to answer any questions. Thank you very much for your attention.
Thank you, Dr. Suresh. That was quite interesting for us. And like you gave many new information, which were like we are mainly from we mainly see the patients. Most of us are medical doctors. So you gave us really insight into the animal thing also, and about the new host of SARS-CoV-2. As is customary, usually we don't ask question at the end of keynote. Since it is a keynote lecture, we don't ask questions. So thank you very much. Now we move forward, and I will now like to invite chairpersons for our next session. And I will also take this opportunity to thank our chairperson for the session, Professor Lalit Dar, and Dr. V.L. Sherwal, Dr. Chand Vattal, and Dr. Rama Chaudhary. I'm very thankful to all of them. They all are great virologists and very busy, but they took time out of their busy schedule and helped us in this. So I'm very thankful to all of them. Now we move forward to the next lecture by Professor R.K. Ratho, who needs no introduction. And I invite Dr. Lalit Dar, Dr. Purva Mathur, and Dr. Malini Kapoor, who are the chairperson for the same. So Dr. Lalit Dar is sir, are you there? Dr. Lalit Dar is a professor.
Yes, thank you so much. I'm here.
Thank you, sir. Thank you. He's a professor in the department, he's a well-known virologist, and he's a professor in the All India Institute of Medical Sciences, New Delhi.
Just finish this 5 minutes thing. Then I get that.
Mathur is a professor in ethics from AIIMS, New Delhi. And we also welcome Dr. Malini Kapoor, who's a professor in Safdarjung.
Yeah, I'll just. Hello, Dr. Ujjwala. Yeah.
Yes, madam.
Yeah. Am I audible now? Yeah.
Yes, madam. Yes.
Yeah. I'm there. Yeah.
Please. And I also invite Dr. Professor Ratho. Sir, are you there? Yes. Yes, please.
So thank you, Dr. Ujjwala. We will invite Professor R.K. Ratho, a good friend and an eminent virologist, now to start his talk on trajectory
I
of...
also invite Dr. Professor Ratho. Sir, are you there? Yes.
Yes,
please.
Yes. Hello, Dr. Lalit. Hello.
Yes, I'll just quickly introduce him, although Dr. Ratho needs no introduction. Professor R.K. Ratho, a good friend and an eminent virologist, now to start his talk on trajectory
I also invite Dr. Professor Ratho. Sir, are you there? Yes. Please.
Yes. Hello, Dr. Lalit. Hello.
Yeah. Yes, I'll just quickly introduce him, although Dr. Ratho needs no introduction. Professor R.K. Ratho, a good friend. So Dr. Ratho, I think there is some lag in the time. Maybe you can start your talk. You really need no introduction. Dr. Ratho is a professor and head of virology at PGI Chandigarh. And he has the he's a fellow of the National Academy of Medical Sciences. He has been awarded the S.C. Agarwal Oration Award. He's a fellow of the European Society of Clinical Microbiology and Infectious Diseases. And he heads his laboratory, which is a very important laboratory. It's a regional lab of the VRDL of the DHR. And the National Measles and Rubella Laboratory and National Vector-Borne Disease Control Program Apex Laboratory. He has many publications and many projects under his belt. Dr. Ratho, please. Thank you.
Hello, Dr. Ujala. Hello.
Hello, welcome, sir.
Yeah. Oh, I think I have sent the video.
Yes, sir.
Please.
Okay, okay. So we'll start. We'll share, sir.
Yes. Hello, Dr. Ujala.
CME COVID-19, Dr. Ujala Ghosal, and the Secretary IVS, Dr. Malik, for giving this platform to me to interact with you in such an important CME on COVID-19. I have been given this task of communicating with the audience about the trajectory of SARS-CoV-2 and its variants during the pandemic waves, which indicates that the COVID-19 variants, how do they prevail over the waves over these 2 years of the COVID pandemic. If this emergent virus appeared to be a novel virus to start with, as per the expected history, there were the appearance of new cases of pneumonia with unknown etiology in the early part of December 2019 in Wuhan city of Hubei province, China. These number of cases, they went on increasing. By the end of the month, there were a series of cases of these pneumonia cases without any etiology. And within 7 days of this the increased number of cases, the first isolate of this unknown virus was identified to be related to a coronavirus. So it was named as 2019 novel coronavirus by the WHO. And within 4 to 5 days of that, there was evidence of this whole genome sequencing, and more so with the first reporting of a mortality. That has created a sensation, and in the end of the January, the WHO has to declare it as an international public health emergency because it has crossed different regions, and also it has reporting of infections among the healthcare workers. That has created a panic situation, and at last, March 11, 2020, WHO declares this as a COVID-19 pandemic. And from March 2020, today I think we are going to complete 2 years of this pandemic, and this is still continuing. Now, when this was looked into, we found out to be that this virus has more genetic similarity with bat and pangolin coronaviruses, crossing over 90% of genome homogeneity. Now, this virus, when you look into the genomic organization, you will understand that this has got a structural component with the N gene, the envelope gene, the matrix gene, and the most talked about is the spike protein gene, which is supposed to be the most important for this coronavirus, for its entry, its pathogenesis, its evading immune system, and even the mutations thereon. And also it is contributed by the non-structural proteins of NSP1 to 16 spanned over ORF1 and ORF1A and ORF1B. And you can think of that these genes, they are utilized in the diagnosis of this coronavirus, molecular diagnosis of this coronavirus, taking either one or multiple genes depending upon the sensitivity and specificity of the testing system. See, the word what was given to me was a project trajectory. Now, trajectory meaning that these viruses have flown like a projectile motion under the action of given forces, and this has traversed from the first wave since its beginning of March 2020 till March 2022. It has crossed 3 successive waves, and it has spread all through the world with it's a lot of morbidity and mortality. Now, if you look into these waves, the first wave which started in India, I think in March 2020, and continued till September, October, November, and you can find the second wave started from the March again to 21, and it lingered till November 2021 with a little gap. I think from January 2022 we have the third wave, and this way if you see, the second wave has got lot of mortality and morbidity in comparison to the first and the third wave. Yeah, this is very well depicted in this graph. You can say the 3 successive waves, and it has traveled all through the world and giving increased number of cases of the coronavirus 19, COVID-19, with the highest mortality during the second wave and followed by the minimum in the third wave. Now, why at all it happens like that? Any virus, it has to percolate, needs to have its sustenance in the community, in this circulation. So this always happens whenever the original virus starts mutating, because due to the original virus, the host tries to get the immunity to it. So the mutants which come out of this original, they try to pervade in the community. So that way, WHO has classified different variants into different groups, like the variant of interest, variant of concern, and variant of high consequence. These 3 categories are being created depending upon their infectivity, the spread, then the disease potentiality, then neutralizing capacity to the previously existing antibodies, either due to natural infection or due to vaccination. So if you look into that, once the variant comes, then it is put into the group of variant of interest. And once it is found to have high public health concern, high mortality, disease morbidity, and hospitalization, death, so vaccination failure, in that situation, they are transferred to the variant of concern. And that in that way, we have, as of now, the B117 alpha, B1351 beta, B1617.2 delta, and then B11529 omicron. These are the important variant of concerns as of now. It has traveled through. But luckily, we don't have any variant of high consequences. If that happens, then it would be a very serious concern. Yeah, if you look into this picture, you will find that the alpha waves, then the alpha waves, then the beta waves, they almost they circulate in the minimum level, whereas the delta one, it has almost percolated majority component, and now that is being replaced by the omicron, which is BA1 and BA2 sublineage. It is still continuing at this moment in various parts of the world. So if you look into this, what is that? This alpha, beta, gamma, delta, and omicron, these are all named because of different mutations. It has come up with the original COVID-19 virus. So the first it came out to be in say October 2020 is a UK variant with 23 mutations. Now this variant has a signature mutant of N501Y in RBD domain, receptor binding domain, then deletion mutant of 6970 and P681H, which is next to the purine cleavage, which created a sensation because purine is an important component in pathogenesis, and if it is affecting that, then it will be more disastrous than what was expected, and It also resulted into increase in R0 value of 0.4, giving a 50 to 70% more transmissibility, viral load with fourfold increase, and then it was 60% of the UK people that required hospitalization in that. And what interestingly, there was no higher rate of reinfection or substantially reduction in the vaccine efficiency with this alpha variant. Then we have the beta variant, which was known as South African variant, and it had 17 mutations and 9 in the S protein. That is what more important, and 3 in RBD. And the important aspect of this is that it has got a higher binding affinity to the human spike protein receptor ACE2, and it has got a capacity of escaping 2 major nutritional antibody against the RBD and one with the loop. Possibly that has resulted into the higher risk of reinfection, as well as half of the persons who had infected didn't develop any neutralizing antibodies against this virus. So we had now the Gamma, which also was known as Brazilian variant, and it has 17 unique mutations with signature mutations of the 501, 484, and then KA417. So this is what is very important, where it has led to significant decrease in neutralizing antibody against the convalescent plasma, and it has also had a higher reinfection rates. The more concerned was the Delta, which was started in India with its signature mutants, and then it had a 10 to 60-fold reduction in neutralizing capacity and lot of breakthrough infections. In addition to that, when K417N mutation was added to it, it came out to be as a Delta plus variant, and it had lot of problems like immune evasion property, increased hospitalization with high requirement of oxygen and ICU admissions, and also it was with the increased mortality and spread all through the world, leading to one of the major pandemic in COVID-19. And lastly, we have the recently circulating Omicron variant. And the interesting aspect of this is that it has more than 50 mutations in various components of the virus, where more than 30 mutations were found with spike protein and other mutations also in different other genes. But this virus has, interestingly, though it has got lot of transmissibility, highest transmissibility, infection rate was high, more of replicability, but it doesn't have much of mortality. So that was the consolation point with the Omicron, and the Omicron variant with R346K, seemingly it appeared to be that it has bypassed the nutritional capacity of the antibody, so possibly it has flattened the antibody response. Thus, we have, in comparison, if you see, we have the alpha, beta, gamma, Delta and Omicron. All these things have almost circulated all through the world, and at this moment we have this Omicron going on in various components. So this graph is going to tell you that how the Omicron at the last, from January to March of this year, is just overshadowing the whole of the world, replacing the previously existing Delta in majority part of the world. So why at all these mutations they do occur? There are few possible reasons which will have worked upon it and shown the evidences. One of these is that high rates of replication and transmissibility of the virus. Like if you see this, to start with, the virus was D614, and when it got mutated to like D2G mutation in the spike protein, it has led to the increased number of cases, and it has replaced the D614 of the original one in the latter part of the April, May, June, July of 2020. So that indicates that a mutant has a capacity to replace the original one and started spreading it. So that was the first thinking. And second is that if there is a higher viral load and if the virus persists in an immunocompromised patient, then what happens? If you take an example, a case when the virus persisted for 190 days, then you can see that there are many mutations accumulated over this period. So these mutants, some of these mutants possibly make the virus to survive within the community, within the host, and it helps in circulation of the virus. And you see the graph where it has been seen that a patient with HIV and SARS-CoV-2, as long as there was HIV virus, the viral load was high, the SARS-CoV-2 was also persisting. The moment the HIV viral load came down, the SARS-CoV-2 viral load also came down. So that it indicates that possibly the intra-host evolution in persistent infection has led to the evolution of these mutants. Now, we also know that during the second phase, we had lot of convalescent plasma monoclonal antibody therapy. So, in presence of this antibody pressure, if you see when it was in vitro test tried with the convalescent plasma and virus kept for 100 days, it has been seen that in various point of time there were a lot of mutations accrued on the virus, and some of them were lethal to the virus, some of them may be helpful for the virus. So thus it has given an idea that in continuous presence of antibody pressure, the virus starts mutating it. And lastly, we had the drugs. When the drugs were started, like the remdesivir, the repurposed drug candidate for COVID-19, its active principle, when it enters into the body, when the infection is percolating, then you can see that this virus, it acts on the RDRP region, it inhibits the RNA replication so that the virus doesn't replicate within the host. So if there is a mutation of phenyl to leucine at 480 or valine to leucine at 547 has led to change in its structure of the spike protein, which possibly has not rendered this remdesivir to be acted upon to that. So in this type of situations, the mutants, they come out and then they start spreading into the community. So thank you very much. These are the possible things, what I can think of about the circulation of these mutants in various waves of the COVID-19 pandemic over these 2 years that is given to us. Thank you very much.
Hello.
Thank you, Dr. Rathod. Very illuminating talk and very comprehensive one too. I'll now ask the organizers to proceed as they would like to. Dr. Ghoshal. My co-chairs, in case they wish to ask anything or give their comments, please.
Yeah. Am I audible? Yeah. So what are the clinical, immunological and the therapeutic implications for these variants in short, sir? Dr. Rathod, sir?
Hello.
Hello, sir, can you hear me? Hello, sir, can you hear me? I'm Dr. Malini this side.
Yeah.
I wanted to know, like, for the audience, like, what are the clinical, immunological and therapeutic implications?
Hello. Hello.
Hello. Hello, sir, can you hear me?
Yeah, you wanted to know the clinical and therapeutic implications of these variants.
Yes,
right?
sir, in short,
Yeah,
for the
in short,
audience, yes.
Yes, yes. Basically, you see we had these alpha, beta, gamma, delta variant and Omicron. If you look into that, alpha, beta, they had the clinical significance, they had increased hospitalization, death rate was not so high when you compare that with the delta 1 or delta variant. Majority of cases, majority of hospitalization, severity of disease, more of oxygen requirement, more of like ICU bed requirement, and more requirement of serum therapy, as well as the medication, whatever required, all were tried during this second wave and the delta variant that had all these things. But if you look into that, the latest one, the Omicron had lot of transmission, then lot of morbidity. People have affected, but it didn't have mortality or severity in comparison to delta or previous alpha. Now, second thing is that possibly people, they think maybe this Omicron is not being neutralized by antibodies, but the immunity what has lasted refers to all these alpha, beta, and delta, and natural infection as well as some vaccination what you have given. Possibly somewhere it has given a way out for this Omicron to remain in a less severe form. That's what it appears to be in nutshell. And second part is
Thank you, sir.
Omicron is also going on. Possibly it is giving a natural boost to the population, so that we Indians feel that we are more immune now than before.
Thank you, sir.
There is one question in the chat box about which says, what about the new variant in China? I'm not sure what they're referring to. I think as of now, Omicron is the predominant one, whether it is the BA1 or BA2 lineage.
Yeah, I think in India we had more of BA2, and now I think outside whatever the Omicrons are coming, they are mostly of BA2. So that way we feel that because we have already exposure to this BA2, we might not have any problem even if it comes from outside if that's strain. So that way we have a little bit of confidence.
Yeah. Well, just one quick question before we close your talk, Dr. Ratho, which was a very interesting one. You mentioned 4 possible reasons for the emergence of the mutants. But we do know that RNA viruses as such intrinsically have the ability to undergo mutations.
Yes.
Do you feel that, in all those 4 causes, which one do you think would be most important, or is it just the intrinsic nature of the virus to evolve even under the lack of immune pressure or the lack of drug pressure?
Yeah, for all RNA viruses, we don't have a drug pressure.
Right.
That is the last possible thing.
Yeah.
Second, the immune therapy or the pre-existing antibodies also a component. It is again it is second to last. So it appears to be that the mutation because of replication property and RDRP proofreading deficiency, that gives the different variants. Now, which variant will come up as a clinical significant or which one is going to have it, that is a property of the virus and the host, how to react to it.
The speed of spread, etc. I think all those things will determine. And just one last point, after Dr. Suresh's talk, I think we need to also draw attention to recombination events, because I went back to our basics, our basic Bible fields virology. And it says very clearly there that coronaviruses are the most likely of the RNA viruses to undergo recombination events, because there is template switching that occurs there. So do you feel that in the long term, maybe recombination events might have a role to play also?
Yeah.
Apart from point mutation.
Definitely. Whatever you have given a point, it needs to be really kept in mind. But this can be possible.
Thank you so much, Dr. Ratho. If there are no further questions, I don't see any in the chat box. I think about the variant in China, they have added that they're talking of the stealth Omicron. So I guess it's still Omicron. Thank you so much. Wonderful talk, and thank you for joining us. I'll now pass on to Dr. Ujjwala Ghoshal, please.
Thank you.
Thank you, Dr. Ratho. Thank you, Dr. Ratho, for an excellent and very lucid talk. Thank you.
Thank you, Professor Ratho. Thank you all the chairpersons.
Thank you, ma'am. Thank you, Dr. Lalit.
Dr. Malki, Dr. Pudwa. Thank you all. Now, our next talk is by Dr. Ellison A. Kelvin, and she will, for this session, the chairpersons are Professor Vijay Lakshmi Nag. She is Professor and Head AIMS Jodhpur, and also a member of National NMC. And then we have Professor Dr. Ravi Shankar, who is a professor and from Professor and Head Biochemistry and Structural Biology Division, CDRI Lucknow. And we also welcome Dr. Suruchi Shukla, she is from Assistant Professor from KGMU and has a lot of work in COVID-19. So I welcome you all.
Good morning, Dr. Ujjwala. Good morning, all. Can you hear my?
Yes, yes.
Can you hear me?
Yes,
Okay. Good morning to all. Thank you, Dr. Ujjwala. Whatever the little information I have about Dr. Ellison Kelvin, She is a virologist at Vaccine and Infectious Disease Organization at University of Saskatchewan, Canada. Her work is focused on emerging viruses and the development of vaccines for these viral threats. Dr. Calvin is an active member of the WHO Ad hoc Advisory Committee of Canadian Society of Virology. She is PhD and she is currently involved in the evaluation of the development of COVID-19 vaccines for Canada. Today, she is delivering her talk on evolutionary plasticity of coronavirus and future evolutionary pattern of SARS-CoV-2. Dr. Ravi Shankar and Dr. Suruchi Shukla, would you like to tell something about the Dr. Elson, or we can hand over to Dr. Calvin.
Good morning, everyone. I'm Dr. Suruchi, and we are thanks to Jula ma'am for this great initiative with the ASM, and we were looking forward for these kind of CMEs and seminars. And Dr. Ellison needs no introduction. Ma'am Dr. Nag has already introduced her so well. So I think we can start with the session. Thank you, ma'am.
Hello.
Hello.
I sent in my video as well.
Yeah, so would you like to share your screen, or should we start from, like, we have your recording? So should we play?
You can, yes, please. Thank you.
Okay. Thank you so much. Thank you so much.
Thank you for having me. Hi, thanks very much for having me. And I checked my email one last time. And the SARS-CoV-2 virus and vaccine strategy. Today, I'll be talking about the intersection of antigenic evolution and the SARS-CoV-2 virus and vaccine strategies, really addressing where do we go from here. First, I'd like to acknowledge that I'm coming to you from Treaty 6 territory and the homeland of the Métis. So like many of you, I first came into contact with SARS-CoV-2 through a notification. And this was a ProMed Mail email that I received in the late hours of December 30th into December 31st. I was about to go to bed and I checked my email one last time, where at the top of my inbox, there was a notification describing the hospitalization of about 30 people due to undiagnosed pneumonia, probably caused by a new virus. Of course, this virus went on to be identified as the SARS-CoV-2 virus. And this virus was quickly sequenced, and properties about the virus were identified, including that this virus was capable of human-to-human transmission, which was publicized on the 20th of January, 2020, leading to initiation of what's referred to as the Great Vaccine Race, where companies and academic researchers at universities began developing vaccines using several different vaccine platforms, such as proteins and viral vector-based vaccines, as well as nucleic acid-based vaccines. But what were they targeting? So analysis of the SARS-CoV-2 virus identified 29 proteins being expressed throughout the genome, but only the S protein or the spike protein located on the surface of the virus envelope was found to be immunoprotective. This is due to the spike protein being responsible for binding to the ACE2 receptor on the surface of target cells, which after binding allows the virus to enter the cell, leading to virus replication and virus dissemination. Specifically targeting this spike protein on the surface of the virion by antibodies can block this process, thereby blocking viral infection. Identifying the S protein or spike protein as the Achilles heel of SARS-CoV-2. Although there are many other epitopes found throughout the genome, such as in the membrane, nucleocapsid protein, or envelope protein, only the S protein so far has been shown to really be immunoprotective. So by targeting the S protein, the mRNA-based vaccines pulled out ahead in less than a year. Clinical trials which finished from Pfizer and Moderna showed that their mRNA-based vaccines targeting the spike protein of SARS-CoV-2 were over 90% effective in individuals who had received 2 doses. Unfortunately, around the same time, less than a year of our time with the SARS-CoV-2 virus, significant variants were identified. These went on to be categorized as variants of concern. The WHO decides variants of concern to be those that have increased transmissibility or a change in COVID-19 epidemiology, increased in disease severity or difference in presentation from the original SARS-CoV-2 virus, or that the new variants can somehow evade preventative measures such as vaccines, antivirals, or social preventative measures. Specifically, we have a couple of variants of concern that are noteworthy, although, however, only 2 are currently identified to be circulating throughout the world by the WHO. Specific variants of concern that we've been investigating include the Alpha, Delta, Beta, Gamma, and now Omicron variant, which was identified at the end of 2021. Several noteworthy mutations were found within the S protein, which of course raises questions on the effectiveness of the vaccines that we had in place. Specific mutations that have been shown to be highly consequential include those in the RBD or the receptor binding domain, specifically N501Y, K417T, and E484K. And this constellation, this triplicate of mutations has been shown to increase the virus's attachment to the virus, as well as associated with increased viral replication and transmission. So once a variant is identified, there's an immediate need to characterize how these mutants will do against our vaccine-induced immunity, which we've already established. Similarly shown in the graph on the right, we can perform virus neutralization assays using serum from people who've recovered from COVID-19 or people who've been vaccinated, and determine how effective their antibodies will be at blocking the virus from infecting cells. Here we see progressive decrease in antibodies from people who've been vaccinated to block variants of concern infecting cells, where Omicron has the most significant decrease, although not always completely ablated. Something else that's important that has come up in the past couple of months is shown that a 3rd dose or a boost of the mRNA vaccines can increase cross-reactive neutralizing antibodies. Here we see neutralizing antibodies in people that had 2 doses of a vaccine against the Wuhan virus, which increased after a third dose or boost. And this was similarly shown where a third dose or a boost increased neutralizing antibodies to similar levels as seen in the Wuhan virus, but for Omicron, where there were previously a very low level of neutralizing antibodies in these individuals before boosting. So now we come to the question, when are new vaccines needed and how should we determine when vaccines should be updated? As this diagram shows, we have pre-existing antibodies elicited through vaccination to the spike protein, but mutations may cause our neutralizing antibodies to be not as effective at binding, therefore leading to escape viruses and possibly more mutants. So it's important that we address this issue. Looking at a related respiratory virus, the family of influenza viruses that cause epidemics each year and sporadic pandemics, we have the WHO's Global Influenza Surveillance and Response System, or GISRIS, where there's 144 national influenza centers over 114 countries that analyze samples in their throughout their epidemics of influenza each year, and this guides the updating by the WHO for the vaccine components for the northern and southern hemispheres. So what questions are they asking, and can this be useful for what we're seeing with SARS-CoV-2? The questions that are asking as the WHO is updating recommendations for influenza vaccines are: which circulating viruses are making people sick, so circulating variants or strains of influenza? Which viruses are predominant prior to the next upcoming season? So this is going to be important for determining effectiveness. How effective last year's vaccines were against the circulating viruses of that time, and the ability of the vaccines to produce cross-protective immune responses against the viruses that are circulating or predicted to be circulating in the upcoming seasons. So how can we take this information and move on to what's next for SARS-CoV-2 variants? Well, SARS-CoV-2 mutation rate has been estimated to be 10 to the -3 substitutions per base per year, which may give us insight into how often we should update the vaccines. We know that although SARS-CoV-2 was thought to have a low mutation rate, we now have seen substantial mutations in this virus, but we ask the question, will this virus continue to have huge mutational changes, or will it become more stabilized as it becomes more human adapted? We know the number of mutant viruses is really related to the number of people infected. So more people infected at a given time will lead to more variants and more mutant viruses. And another question that's important, will variant phenotypes become more or less severe? We've seen them both. But what's really important that we all need to recognize is that a surveillance system similar to what I just spoke about with influenza viruses is needed so that we can identify emerging variants of concern. So as we move forward, how do we update these vaccines? Do we continue to have Wuhan-specific boosters, or do we create more seasonal variants of concern vaccines similar to what we have for influenza? And with this, if we update the vaccines, do we pool sets of variants of concern sequences for multivalent vaccines, or do we have more pan or broadly reactive vaccine approaches based on conserved epitopes such as the S2 region of the S protein, which is more highly conserved. And how will this affect effectiveness moving forward is a question we'll need to be asking for each one of these platforms. Vaccine regulatory questions are also important. How do we optimize the sequence of our booster programs? Do we do this every 6 months, every year, every 2 years? How do we incorporate mix and matching? We know most countries have favored mRNA vaccines based on problems with boosting of viral vectored vaccines. As well, when should we implement clinical trials for updated SARS-CoV-2 vaccines? And how do we set the bar or determine what the endpoint of these clinical trials are? We know most people now have pre-existing immunity to SARS-CoV-2, so we need to decide, are these new clinical trials the endpoint prevention of transmission, symptomatic disease, hospitalization, or association with death? Another important aspect to consider is how our pre-existing immunity is interacting with updated vaccines or new variants as they emerge. We know that your pre-existing immunity can have significant impacts on vaccine responses, as well as your outcome of new infections. This has been shown specifically for SARS-CoV-2, where people who had Wuhan or Alpha virus as their first infection had differential outcomes after vaccination, where essentially people who were infected with Wuhan had more cross-reactive antibodies after they were vaccinated compared to people who had Alpha and then were vaccinated. We also need to recognize that adding variant vaccines will add more diversity to people's pre-existing immune responses, and effectively we're playing catch up to the virus if we just update vaccines yearly based on the variant of concern that's predominant, and that this might lead to vaccine escape mutants. So in conclusion, I do want to point out that new vaccines will be needed because new SARS-CoV-2 variants will continue to emerge. However, we don't know if these will be major changes or small changes. Our current COVID-19 vaccines are still effective, but if we have major variants of concerns emerge, they might not be effective for long. The S protein is our major target because it's immunogenic as well as immunoprotective. And as well, we don't know the durability of the current vaccines. So how we should update should also play into, take into consideration how long our memory response is. As well, there are several new vaccine strategies that we should consider: multivalent, targeting conserved epitopes, or targeting alternative viral proteins, which I didn't talk about here, but might be a basis for a T-cell based vaccine. And how should vaccines updating be determined? Do we need a calculation for antigenic distance between our previous vaccine and the now circulating variants? And how should pre-existing immunity in the population be considered? So thank you very much for listening.
Persons. Thank you, Alison, for a wonderful talk. Over to the chairperson. Do we have any question in the chat box?
No.
Okay. Dr. Vijayalakshmi, Dr. Suruchi, are you there? Mute नहीं कर दो. Unmute them. Dr. Vijayalakshmi, Dr. Suruchi, are you there?
Yes, ma'am. Yes, ma'am. Yes. So, Dr. Ellison, very nice presentation, ma'am. And we had a lot of confusions and still confusions in the mind regarding sequencing of the COVID. So as we seeing the current scenario in India, the COVID is going down. And do you think really we should, at the public level, we should continue the sequencing, or we should go back to the same system of the influenza just for the vaccine strategy we should go on? And another question which I was in my mind, that we have so many sequences, Omicron, Delta, and we had vaccinated time to time. How this messenger RNA, do you think any messenger RNA vaccines, why these reinfections? Could you show some light on this? Why this reinfection apart from so many? But this is the first question which we receive. We have received vaccines, but we are having a reinfection. So can you show some light on this? Thank you, ma'am.
Sure. So I think right now, because we're not in a stable epidemic pattern as we are with influenza viruses, we really do need to keep sequencing and keep testing so we know what the viral burden is within the community and what exactly those viruses are. And that way we can prevent possibly new variants, but also plan for what is circulating. In relation to your other question, I think it's multidimensional. When what we know now, what we didn't know before, is that vaccination with mRNA-based vaccines do seem to have not the longevity which was initially predicted. So you will have decline of neutralizing antibodies that might play a role in the ability to be reinfected, but also the change in the antigenicity between what's circulating and what people were initially vaccinated with is probably also playing a role. So I think we have to look at those factors. And then additionally, when you get a vaccination within your arm, so intramuscularly, it's mostly providing protection to your lower respiratory tract and not so much to your upper respiratory tract in your nose, which can allow, and we know this from influenza vaccines too, that when you get your intramuscular vaccination, you can still get infected, especially in the upper respiratory tract, which will lead to, unfortunately, transmission events, but it will decrease the severity of the disease that's associated with each infection. So I think, it's again, a multi-dimensional problem that different vaccine platforms are trying to address right now.
Yes, ma'am. Yeah, very rightly said. Thank you, ma'am.
Dr. Ravi Shankar, you would like to say something?
Yeah, so this has been a pleasure listening to Dr. Allison Kevin. One other question which always everybody wants to know is the level of protection in terms of time duration against future variants. So what is your take? This is like a layman's question, but it's something that everybody keeps asking all the time.
You mean what's the duration of protection from vaccines with future variants?
Yeah, sure. That's exactly.
Yes, I unfortunately am very bad at predicting what the next variant will be. Omicron really changed the way that I looked at SARS-CoV-2, and I was surprised at how many mutations that it had compared to the previous variants. So at this point, and I kind of discussed it in the talk, that I feel that it could go either way. We could see new variants that have decreased numbers of mutations as the virus might be settling into being humanized and being a human virus. As we heard earlier, a fabulous talk about how the virus does spill over from the animal reservoir. some virologists think that it will become more humanized and more stable to people and have less mutations with less associated variants. But then other virologists do suggest that we can get these new variants with large numbers of mutations through several different pathways, which was discussed in the talk previous to me and done very well. So I actually think both are possible. What the frequency of those will be, time will tell, unfortunately.
Thank you. Since we are running late by 15 minutes or so, so I would, and it is a request, it is quite late in US and our next speaker is from there. So I would last question in chat box. Dr. Vijayalakshmi, please, there's a chat box.
If there are any questions from the forum? Yes.
Yes. I can see a question in the chat box that earlier mRNA vaccine did not work well. Then suddenly what changed? How come they are effective now? Now we have good mRNA vaccine. That is the question.
So I guess the development of the mRNA vaccine platform has had several substantial developments in the last couple of years. One is just with the antigen identifying how to make the spike protein more stable and not dissociate into the S1 and S2 domains, as well as the lipid nanoparticle allows the mRNA to be protected through delivery. So the first mRNA-based vaccines, the mRNA was quite susceptible to being degraded and not actually having the payload delivered to where it needed to be, as well as other stability pieces in the design of the mRNA itself.
Thank you, Dr. Ellison. Thank you all the chairperson for your active participation. Now, before we move on to the next session, there will be a short break of just 1 minute. Thank you all.
Thank you very much.
Thank you, Dr. Calvin and Dr. Ujjwala. Thank you.
Dr. Suruchi and Ellison. Thank you all. All the participants are requested to put on their videos for a picture. We would like to take a picture of yours. Please switch on your cameras. Everyone, please switch on your cameras for a nice picture. Please take whatever is possible. Go on. इसका presentation था. Issues. Thank you all. So I have great pleasure in introducing our next speaker, who is Dr. Swam Prakash. He is our own alumni from SGPGIMS and now is working in US. And I would also, here is I would also like this opportunity to thank my PhD student, Dr. Prabhat Ranjan, who is a close friend and was instrumental in bringing him here. So welcome, Dr. Swayam Prakash. Am I audible?
Yes, ma'am.
Dr.
Thank you
Swayam,
for
can you
having me.
listen? I would.
Am I
Yes,
audible?
I would. Yes, yes.
Yes,
The
Dr. Prakash, you're audible. Okay.
The chairpersons of this session are Dr. Jyotsna Agarwal, who is a professor and head Department of Microbiology, RML Institute, Lucknow, and old friend. Then we have Dr. Manodeep Sen, who is also a professor in the same institute. So over to Dr. Jyotsna, and then we also have Dr. Shailin Saxena, who has been introduced early morning. So over to the chairpersons. Dr. Jyotsna, Dr. Manodeep.
Yes, ma'am. So good morning, all of you, and good evening to our overseas speakers and audience. First of all, I would like this to take this opportunity to thank the organizers in general and Professor Ujjwala Ghoshal ma'am in particular for inviting us to chair this session on working down the COVID-19 pandemic through a pan-coronavirus-based vaccine approach, aiming at all current and future SARS-CoV-2 variants. This will be taken up by the illustrious Dr. Swayam Prakash. He is a postdoctoral fellow from the Cellular and Molecular Immunology Lab, Gavin Herbert Eye Institute School of Medicine, University of California in the USA. He has a specialty and expertise in transcriptomics, immunology of infectious diseases, molecular genetics, genomics, bioinformatics, and computational biology. So over to Dr. Swayam Prakash for his lecture. Can you continue?
Thank you, sir. So, ma'am, that video is. Did you receive that video, or do you want me to present?
We will share. Yes, we received, and we'll share that.
Okay.
Thank you, sir. Thank you very much, and welcome.
Welcome. Please check if it is not proper, then I can present. Just let me know.
So I hope it will work. Just give us a moment.
Good morning, everyone. Thank you, Professor Ghoshal, for having me. So I shall be presenting on our vaccine approach towards the development of a preemptive pan-coronavirus vaccine here at the University of California, Irvine School of Medicine. So the SARS-CoV-2 has created a global pandemic response responsible for millions of deaths and untold morbidity and unknown long-term consequences to human health and the global economy. Success of the current vaccines has been remarkable in limiting the scope of the pandemic, but is not a long-term solution for addressing the current rise of virus variants or for preventing future outbreaks. So we have identified a novel and innovative vaccine approach to create a superior next generation preemptive coronavirus vaccine capable of eliminating the current outbreak and preventing the reappearance of previous circulating SARS-CoV or the appearance of the future new coronavirus outbreaks. So currently there exist several COVID-19 vaccines which appear to be safe and effective in large-scale efficacy clinical trials and have been granted emergency use authorization. Although 2021 was the year of COVID-19 vaccines, but it was also the year of variants. Researchers identified multiple variants of concern beginning from late 2020 to late 2021. And researchers are eventually literally racing these days to determine how different vaccines will hold up against the first spreading Omicron. And a preliminary study recently came up from the UK that is suggesting that at least 2 vaccine doses offer a little protection against becoming infected with the Omicron. And researchers expect that vaccines will continue to prevent severe disease caused by the variant, but to what extent it is not clear. So in line with previous studies, individuals that were recently vaccinated with Moderna or Pfizer achieved substantially higher wild-type neutralization titers than the AD26 specific vaccines. And we observed that individuals vaccinated more than 6 months prior exhibited substantially lower but mostly detectable wild-type neutralization. And prior history of infection was associated with high levels of wild-type neutralization titers, even in distantly vaccinated individuals. Specifically, we observed in the AD26 specific vaccines. So given the drastic increase in cross-neutralization of the SARS-CoV-2 Omicron pseudovirus in the boosted versus non-boosted vaccines, we directly compared the sera from individuals that recently received their primary series to those that were boosted with an mRNA vaccine within the last 3 months. And wild-type pseudovirus neutralization was comparable between individuals who received 3 versus 2 doses of either the mRNA vaccine. Keeping these things in mind, we came up with certain strategies to address the existing risks. So, although we have the option for the periodic booster immunization with the current vaccines, but this is a short-term solution at the best, and there is a risk of T-cell exhaustion with repeated boosting of the spike-only vaccine is still there with the current vaccines. And we are looking forward to a preemptive vaccine eliciting broad-based immunity on targeting conserved antigens. And this is based on the growing evidence for important role of cross-reactive T cells with protection from the COVID-19. So it is really difficult to update the vaccines, keeping in view the rate of evolution, the number of mutations the virus is coming up with. And it is really difficult to come up with novel vaccines specific to different variants, and that is not a very economical solution. So the current vaccines address 2 key gaps. Our vaccines. Our vaccines usually address 2 key gaps and limitations which are there with the existing vaccines. The first hurdle is the first gap with the current vaccine is the immune responses are directed only towards the spike antigen, whereas the vaccine which we are coming up with, it includes more conserved antigens along with the spike antigen. So the current vaccines focus primarily on the induction of antibody responses, and which misses the opportunity to target long-term cross-reactive memory T cell responses. And we are actually we have identified antigen sequences that are highly conserved and common to nearly 9 million of the human and animal coronaviruses. And we eventually came up with 7 prototype pan-coronavirus vaccines, which I'll eventually show you. So a brief overview of how we identified these conserved reasons. So before going to that, it is really remarkable to see the rate at which the virus is evolving since 2019. So there are almost 23 lineages now of the SARS-CoV-2 since December 2029, and this comprises of hundreds of non-synonymous mutations spread across the structural proteins as well as across different non-structural proteins of the SARS-CoV-2. So why we keep this in mind while screening for the prototype pan-coronavirus vaccine? Because this is really of importance. If we will be able to include the conserved mutations into our vaccine constructs so that the vaccine will provide sufficient immunity from not only from the current or the past variants, but also from the future variants of interests or variants of concerns. So in the first approach, we identified 5 conserved regions of SARS-CoV-2 genome, which spreaded across 7 conserved antigens, including the spike glycoprotein, nucleocapsid, envelope membrane, and several non-structural proteins, including the ORF1ab, ORF7, and ORF3. And the second approach was to identify the highly conserved CD8 and CD4 positive T cell epitopes. So determining the antigen and the human B cell, CD4 positive and CD8 positive T cell epitope landscapes that are conserved among the human and animal coronaviruses should inform in the development of future pan-coronavirus vaccines. Using robust immunoinformatics approaches, we have identified several human B cell and CD4 positive and CD8 positive T cell epitopes that are highly conserved in about 8.9 million SARS-CoV-2 genome sequences identified across the globe. 6 circulating coronaviruses that caused previous human outbreaks of common cold, SARS-like coronaviruses isolated from bats, pangolins, civet cats, and MERS strains isolated from camels. So notably, we found that the identified epitopes actually recalled the B cells and the CD4 positive and CD8 positive T cells from both COVID-19 patients and healthy individuals who were never exposed to SARS-CoV-2. And this also induced strong B cell and T cell response to humanized HLA-A2-DR double transgenic mice. The findings were very much motivating and paved the way to develop a preemptive multi-epitope pan-coronavirus vaccine to protect against, like I said, past, current, and future outbreaks. So how we did the validation of these vaccines? So next we assessed whether the potential SARS-CoV-2 CD4 positive and CD8 positive T cell epitopes that are highly conserved between human and animal coronaviruses would recall the memory CD8 positive T cells from COVID-19 patients, as well as from the healthy individuals who have never been exposed to SARS-CoV-2 or to COVID-19 patients. So we observed that among the 27 SARS-CoV-2 epitopes, 7 epitopes recalled a strong memory, a CD8 positive T cell response from the unexposed healthy individuals, and 5 CD8 T cell epitopes showed an intermediate response, and about 12 CD8 positive T cell epitopes also showed mild to intermediate immune response. So similar to SARS-CoV-2, memory CD8 positive T cells, memory CD4 positive T cells, specific to several highly conserved SARS-CoV-2 epitopes, were detected in both the COVID-19 recovered patients and unexposed healthy individuals, and their immunogenicity and antigenicity were assessed. And subsequently, we observed significantly elevated T cell responses against conserved epitopes, which were promisingly correlated with better disease outcome. And the immunogenicity of the identified SARS-CoV-2 human CD4 positive and CD8 positive T cell response, epitope-specific response, was assessed in the humanized HLA-A2-DR double transgenic mice, and a mixture of peptides incorporating the CD4 positive T cells and CD8 positive epitopes were delivered with CPG and alum. So in the same light of the results which we observed in humans, here also we identified strong intermediate and low T cell response associated epitopes in the HLA transgenic mice. So altogether, these results indicate that pre-existing memory CD4 positive and CD8 positive T cells specific to both structural and non-structural protein antigens and epitopes are present in the COVID-19 patients and unexposed healthy individuals. Although the SARS-CoV-2 specific CD4 positive and CD8 positive T cells in the COVID-19 patients and healthy donors target epitopes from the whole virus proteome, most T cell epitopes are concentrated in the non-structural proteins, with open reading frame 1AB being the most targeted antigens. So, we observe them to recognize highly conserved SARS-CoV-2 epitopes that cross-react with the human and animal coronaviruses. And we also found that infection with a common cold coronavirus or human exposition with animal and pet-related coronaviruses induced long-lasting memory CD4 positive and CD8 positive T cells specific to the structural and non-structural SARS-CoV-2 epitopes in healthy unexposed individuals. So we next predicted the potential B cell specific epitopes and subsequently looked for their immunogenicity and antigenicity. In the same light, we screened for our CD4 and CD8 specific epitopes. And as expected, the non-immunized animals or those that received here the adjuvant alone did not develop the detectable immunoglobulin G response, and of the 28 B cell epitopes which we screened, so 6 epitopes were found to be of highly immunogenic and 5 epitopes were highly antigenic, and they were recognized by the serum IgG from the COVID-19 patients, confirming the presence of at least one native linear B cell epitope in each of the peptide. So based on all this data from the human and the animal based experiments, we came up with this one of this proposed vaccine map, which comprises of 16 CD8 T cell epitopes and 6 CD4 T cell epitopes and 8 highly conserved B cell epitopes. So the beauty of these epitopes is that when we looked at the population coverage analysis, so about 92% of the global population is covered when the CD8 specific T cell epitopes were taken into consideration, and about 89% of the global population was covered when the CD4 specific epitopes were taken into consideration. So this shows the robustness of the vaccine, and if it will be in use, how efficient it will be keeping in mind the global population. So coming to the animal model validation for the vaccine construct, which we included 16 CD8, 6 CD4, and 8 B cell specific epitopes. So we have further used, like I said, these CD8, CD4, and B cell epitopes to construct the multiple several multiple candidates vaccines using the mRNA adenovirus and adeno-associated virus-based approaches. And the vaccines were screened in triple transgenic HLA-A2-DR human SE2 mice. And the epitopes are used in multiple combinations to select the best vaccine candidate. And we challenged these mice, these immunized mice, to not only to the parent Wuhan variant, but also to all the variant of concerns like alpha, beta, gamma, delta, and omicron, to see how efficiently our vaccine is working against when exposed to multiple variants of concern. So this is a preliminary data, and looking at the weight loss and the survival, it's showing the efficiency of the vaccine when compared to the placebo group, and clearly the Wuhan specific variant and the alpha variant or the beta variant, when exposed to these variants, the immunized mice, they are showing almost they are showing relatively significantly less weight loss, as well as almost 100% survival was there with the Wuhan and the South Africa specific beta variant. And coming to the Omicron variant, also it is really motivating to see that all the triple transgenic mice we included in the experiment, they survived. So which shows the efficiency of the efficiency as well as the robustness of our vaccine selection strategy. And looking at the pathology, also it shows in comparison to the mock treated animals, the peptide vaccine treated and the adeno-associated specific AAV9 specific vaccine specific animals, they showed relatively better pathology and also less disease and better survival in comparison to the mock. So at the end, we performed a single cell RNA sequencing based analysis to see whether how the CD8 T cell specific population is behaving in lieu of our vaccine. In panel A, you can see a comparison between human samples who were human COVID-19 samples who were non-vaccinated and COVID-19 patients who were vaccinated with one of the mRNA vaccine. And in panel B, it is showing our mice treated with the pan-coronavirus vaccine and another set of mice subjected to non-vaccination, but both were subjected to challenge by the Omicron variant. So here you can clearly see when looking at different cell clusters, in the human specific panel, the CD8 specific T cell frequency is relatively low in comparison to the non-vaccinated panel, non-vaccinated individuals, even though people are vaccinated with the mRNA vaccine X. Whereas when we immunize the mice with the pan-coronavirus vaccine, in that scenario, the CD8 T cell response is significantly high, and the CD8 T cell specific frequency is also going significantly up. So based on these, there are certain advantages of the novel preemptive pre-coronavirus vaccine in comparison to the existing COVID-19 vaccines, like it includes the highly conserved virus sequences, it activates the T cell arm both the CD4 positive and CD8 positive T cell arms of the immune system. And this pan-coronavirus vaccine will comprise of the mRNA encoding the conserved coronavirus antigens, and thereby it will provide a broad-based immune response capable of long-term cross-protection against current virus variants and newly arising coronaviruses in the future. So in comparison to the other pan-coronavirus vaccine developers, our vaccine is at very much at a very advantageous position. So it includes the spike protein antigen, it includes the other conserved non-spike specific antigens, includes the neutralizing antibody epitopes, includes the CD8, CD4 specific T cell epitopes, and it targets all the variants of concern, variants of interest, and it includes the protective asymptomatic CD4 positive, CD8 positive T cell epitopes, and it is designated to target past, present, and the future pandemics. So to summarize, our unique strategies addresses the gaps which we discussed at the beginning of the current COVID-19 vaccines. These are more conserved structural and non-structural coronavirus antigens, and it includes the epitopes to elicit the neutralizing antibodies and long-term cross-reactive CD4 and CD8 positive T cells. And the antigen selection was validated by the correlation with positive disease outcomes in human patients as well as in the animals, and protective efficacy against variant strains in the animals. So antigens are delivered by the mRNA platform, validated in humans. And we filed 3 patents within publication of the SARS-CoV-2 specific sequences, conserved sequences which we reported. And patent applications covered the 7 candidate preemptive vaccine antigens, and preliminary freedom to operate analysis has not identified any blocking of the patents. So thank you so much for having me, and if there are any questions, please let me know.
So thank you, Dr. Kamkata, for your enlightening lecture regarding the curbing down the COVID pandemic through this your pan-coronavirus-based vaccine, aiming at all current and future SARS-CoV-2 variants. If there are any questions in the forum, I would like to invite. If there are no questions from the forum,
Is there any questions?
Vanudeep, can you listen? Dr. Swam?
Yes, yes, ma'am. I've already
Do you have any question?
Okay. So, okay. The question from my side is just that, sir, first of all, I would like to thank you for your eloquent lecture, your extraordinary detailed research on a prototype pan coronavirus vaccine, promising preliminary promising observations regarding inclusion of spike and non-spike antigens with neutralizing antibodies, including arming the cellular immunity, as well as targeting variant of concerns, including past, present, and future variant of concern, with clinical validation in humans, as well as having protection against variants in your animal experiments. I would just like to just ask her how many since how long has this research been going on, sir?
So we started right from the beginning of the pandemic. So it's almost 2 years we are into this. So this is one of our vaccine approaches which I presented. So we have actually multiple vaccine approaches, and one of our colleagues, Dr. Pierre, Madam Professor Ghoshal, Dr. Pierre is also here, I think. So he's also going to present probably. Yeah. So we have multiple vaccine approaches, and this is just one such approach in which we have large sequence vaccines in which we have included both the structural as well as the non-structural proteins. And as well as the second approach, which I elaborately discussed, is the multi-epitope pan-coronavirus vaccine. So we are trying to use multiple platforms to deliver these vaccines. For example, we are going for the mRNA-based approach. We are going for the adenovirus-based approach or the adeno-associated virus. And to why so many approaches? Because we don't know at the beginning when we start with a vaccine which approach will work the best, but definitely the mRNA-based vaccine is a robust approach, but still, we are working on multiple domains basically, and it's very much promising to see the current data, keeping in mind how the immunized mice challenged with the multiple variants of concerns are showing protective, are being protected from the SARS-CoV-2. So, yeah.
Thank you, Dr. Swayam, and thank you all the chairpersons for your active participation. Thank you. Now moving over to the next session. Next lecture will be taken up by Dr. Himanshu Reddy. He is additional professor in King George Medical University. And for this session, our chairperson are Dr. Vajanti Mala Mishra, who is professor and head, Department of Microbiology, AIIMS, Bhubaneswar. Then we have Dr. Gautam Palit, who is ex-professor and head pharmacology and director grade research scientist in CDRI Lucknow. And we also have with us Dr. Richa Mishra, additional professor in microbiology, and Professor Sangram Singh Patel, who is associate professor in our Department of Microbiology, SGPGIMS. I welcome them all. Over to them. Over to the chairpersons. Dr. Gautam, Dr. Vajanti Mala, are you there?
Yes, we are here. Good morning. Good morning, all of you. And today the next speaker is one of from our medical college, Dr. Himanshu Reddy, and he will be talking on issues with the diagnosis of emerging SARS-CoV-2 variants, the clinician perspective. I think this is one of the most interesting. We have heard so many interesting topics which covers the various aspects of the COVID and their vaccinations and the virus and all these things. So I will, without wasting any time, I will request Dr. Himanshu to express his view and what is his clinical perspective, and we are eagerly to hear from you, Himanshu.
We wanted to see silence that we had the SRNA and look into effect
virus speaker.
If this is an issue, I can directly present also. Hello and good morning, everyone. I would like to thank the organizers for giving me this opportunity to present in front of you all, and I also would like to congratulate them for coming up with this very different topic. So before I go ahead, I would like to definitely state this thing. This perspective means view, and this is my point of view. So there might be issues which others might have, and I apologize for that at the very beginning. When we talk about perspective, the perspectives can be of different types. The microbiologist perspective, a very important one, that is identification of organism and its strains, basically identifying them, which is very important. You see, when you have new organisms coming up and when you identify, then only you can develop newer diagnostic kits. You can predict whether there can be any pandemics or epidemics, and this is very also important also for developing of vaccines and drugs. Secondly, epidemiologist perspective is something which is more on a social level, where they want to see what is the level of infection in the society, what is the risk for the society, and how we can prevent this risk. Identification of infection for epidemiologists includes finding antibodies, seeing the at-risk population and all. But for clinicians, it's much more pointed, pinpoint identification which we require. We are basically interested in treating and identifying those infections which are of critical, which are severe, and who require urgent intervention to save their lives. So that is what a clinician wants. He wants to identify the patients who are having severe disease or who might land up in severe disease. Now, what are the variants? Well, we know that viruses can mutate and they will mutate. That's the character, and we'll keep on having newer variants coming up every now and then. But not all variants are of major concern, and that is why the United States has a different surveillance system, and it also has systems for actually identifying the variants. And it has a classification. As per the US government classification, there are four classes of variants. Variant being monitored, which now there have been many, but there is none which are continuously going on. Various variants of interest, who might actually have certain concern, who might be having changes in their material DNA material, which I think already the microbiologists here must have explained. And variants of concern, that's what we are interested in, those who have the risk for rapid spreading of the disease, or those who have the risk for increased severity. And they can also be missed out with the present, they might have the risk of getting missed out by the present testing. Then variants of high consequence are those who will cause severe disease and will also skip or slip out of the testing mechanisms and will also not be covered with the present vaccines. So these are what the divisions are. And if you look at it, the most important concern for anything is the evidence of increased transmissibility and evidence of increased disease severity and impact on diagnostic treatment of the reduced efficacy of vaccines. There are different genes which are actually, which actually lead to, in which the mutations occur. And as of now, the two main variants of concern are Delta and Omicron. That's all we are basically interested in. And even as a clinician, this is what we want to look at. And the reason is very simple. Delta we already have faced, which occurred in the first half of 2021, around April, May. It was actually identified from India, where there was a huge number of casualties, and it spread out much faster than the variants which were otherwise prevalent at that point of time, and very rapidly took over and the viral population throughout the world and became the dominant variant. Then at the end of 2021, we started seeing Omicron cases. Omicron first identified in South Africa, and it spread out very rapidly to the limit that it had a rapid peak, and we faced the brunt in end of December to January, and it started decreasing in February. The good thing is the Omicron, though it spread out fast, it did not have the same amount of mortality, which could have been because of multiple reasons, including the prevalence of higher amount of vaccines or the previous exposures, which might have led to some bit of protection, though this hasn't been proven as of now. Having said that, during the Omicron crisis, we as clinicians, we did have this feeling that we might be missing out on cases. Many of the patients who are actually coming up with mild disease might be missed out with the present diagnostic kits. One of the reasons was the change in the spike protein. So what are the changes? You see the changes were, there are different types of changes, which I'm pretty sure my esteemed presenter, the next presenter, Professor Ujjwala Ghoshal ma'am, would be actually presenting about. But here I just wanted to state is the biggest concern for the clinicians was that there was a change in the spike protein. That would mean that there would be difficulty in the present medication that we are using or the vaccines. And also there was this concern that because spike protein was something which was being monitored, and they might be slip out of these cases and not be tested by the present kits. So just to get that to a rest, I wanted to share a little bit from which I actually came to know from my microbiologist colleagues only, that when you do a diagnostic kits in India, which is basically supplied from the ICMR and which has been ICMR regulated kits, you have one screening gene, which is a pan-COVID corona gene, and then you've got two confirmatory genes, and most of the kits that we're using are actually using RDRP genes. So therefore, the chance of us missing out on just because there are mutations in the S gene are very less and unlikely. And if you look at it, we did identify a lot of cases, and therefore the concern for clinicians of missing out was less, even though it was there in the beginning. But what are the issues for a clinician? Well, the issues for a clinician is, number one, missing out on serious infections where you are not so sure, and overdiagnosis of mild disease. Because once you start doing that, then you have this high load on the hospitals, which are already overburdened, which would not only increase the number of COVID admissions, but also restrict the admissions of other diseases who lose out from care, which have to be provided to them urgently. There's another issue which comes up is many of the times patients who are admitted for other diseases, and because of these several protocols which are prevalent, they are tested, and even with mild COVID symptoms, or may not be even, they might be just asymptomatic carriers, and they are diagnosed. And what happens is these patients are then shifted to the COVID wards, which, as we know pretty well, are not similar to what we would expect from what's the sort of clinical expertise and the amount of infrastructure that is present in a non-COVID ward. So this is what we as clinicians are basically a bit concerned about, that the diagnosis should be such that we are only and only treating the serious infection. What are the causes of missing out on the serious infections? One, we have to accept that is the proper sample collection. Improper sample collection, improper method of sample collection. If you take out the swabs at a very tip and not actually go deep down and take them out, then you will miss out on that. And you can find that out if you also look at the human, DNA, which you do that it's done by the microbiologist. And if the human cells are not there, that means they have not been properly scraped out. Then the improper sample transport, which is very commonly seen, not having kept in proper ice box, leaking out. So there are those issues which are prevalent and which we have faced throughout the time, specifically for transporting from the clinical wards to the labs. Late sample collection. Now, patients might have had actually minor symptoms for a certain period of time, but what happens is they start coming to the hospital once these symptoms actually get aggravated, and they forget about the back history. Now, the clinicians would actually go about, and if you are not getting a complete history or you are not taking a full history, then you might just do a testing. The patient might come out positive, and the patient might come out negative because the patient has been tested late. The virus itself might have actually decreased in its load, but the disease has progressed because of the immune-mediated injury, the immune system activation, and you might just not be able to diagnose COVID or not. But having said that, in such an issue, you also have to understand it is basically, therefore, there is no role of the antivirals which you would have to give otherwise in the early cases. And finally, of course, there was preventive COVID, a term which was coined in the second wave, where many patients who had radiological and clinical picture, but their tests were coming out negative. What they were exactly, it's difficult to say, but then you have to understand it's not as if the radiological picture that we see is 100% only and only for COVID. There can be certain overlaps which has to be kept in mind. Do they make a difference if you notice? Yes, it does. We saw in the second wave where there were multiple uses of CT scans to the limit that people were doing CT scans and then they were doing Corrad scores. That was leading to one, many of the times the radiologists were diagnosing it as COVID and the microbiologists were not. And equally then the other issue was that sometimes, other diseases were also being classified as COVID, even though the test results were coming out negative and people were treating them as COVID. It could have been some other viruses. We saw sometimes having atypical bacteria, and of course, even H1N1 came out positive. So these things can happen. So people have to understand that getting a negative COVID test doesn't mean that, and just getting a radiological imaging, that you should start thinking about COVID only. There can be other bacteria and viruses which can mimic it. The most important fallout, of course, was the unnecessary use of steroids because of the fear factor which had actually developed among the clinicians, which of course, specifically in the second wave, and people were using a lot of steroids, even when little bit of changes were there in these CT scans, even when the COVID tests were negative. This led to another major issue, which was the outcome of the mucorrectomy. And finally, of course, there's a dilemma regarding the use of antivirals. But overall, as a clinician, as an infectious disease specialist, and as microbiologist, I think you would agree with it. If the virus is coming negative, then there is no role of any antivirals to be used for such. The overdiagnosis, well, as I've already pointed out, it leads to unnecessary admission, overburden on the healthcare, and deprivation of health facilities in non-COVID diseases, which has a huge impact. We have seen so many patients who should or could have been, directly treated in a non-COVID wards, but just because of, sometimes the reports from outside were positive and they were brought into the COVID wards, kept for a day or two. When repeated, they were found to be negative. But that golden period for those patients, it gets hampered. The treatment, even with our best of the best efforts, it is not the same that you can give. For example, if somebody has an MI and he's in a COVID area, as compared to in a patient of MI in a cardiology place where they can be immediately taken to, for angioplasty or something, or similarly for a trauma patient who is kept in a COVID ward. So these are the issues which one should not look over, because that has made a huge difference for many patients, and that has led to increased mortality among these patients. One important thing is the variants that we see. It's very important that sometimes, as a clinician, we have a different thinking as compared to an epidemiologist and the microbiologist. For example, let's take the three waves. If you look at the three waves, the first wave, the second wave, and the third wave, the clinicians definitely had a huge role in the first and second wave, because look at the wave. For the same wave, the death rate was much less in the third wave. And as a clinician, we are concerned about the number of serious patients who had to be admitted, especially pure COVID or COVID-induced admissions, as compared to just incidental COVID, and the death basically occurring because of that. And in the third Omicron wave, we hardly had that much of admissions as compared to the first and second wave. This has also been seen comparatively even in the data which has come out from South Africa or from other places, and which has been seen in even studied by the CDC, and even recent ICMR studies have shown that the graphs that the death rate was very minimal as compared to what was seen with the first and second wave. So as a clinician, we know that the variance will be the norm, but we always want to know that we have the current PCR tests. As long as we are sure that they will be able to diagnose them, we are pretty comfortable about it. But judicious use of tests should be the norm. one should not actually make unnecessary, protocols which can actually lead to over diagnosis of mild disease, which can lead to unnecessary over admission and unnecessary overload of the healthcare facilities, including even in a similar vice versa. One should also remember that if you are suspecting high risk patients or you are having a severe disease, then of course go ahead, test completely, not just do a antigen based kits which might or may not pick them up. Then go ahead and do the complete panel, find them out, treat them early so that you can decrease the mortality, which should be the goal for a clinician. So with that, I would like to thank the organizers for providing me this opportunity so that I can put out my view regarding this topic.
नहीं आपको क्वेश्चन मिल गया था? हाँ देख ली वो डिस्कशन है ना, पैनल डिस्कशन 1:30 बजे। ओके ठीक, थैंक यू।
Hello.
Hello.
Hello. Hello.
Over to chairpersons.
Dr. Himanshu, it was a very nicely presented, and especially your conclusion is very thought-provoking, and it's a message to the others, the over-diagnosis and testing, and because it has become, it's a household, people start doing it at their houses and then they confirm it. If it is positive, then they hide it, and if they are not. So what you want to say for the, if we have something in the future and what should be done as a clinician, that should be given to the lay person or general, what do you think should be done?
Yes, so thank you, sir. Well, I think we already have a model regarding this. We have seen in H1N1 where we actually test those who are high risk. You basically want to restrict to those patients, test them where you have to actually intervene. Without doubt, elderly, immunocompromised, where they're having symptoms, and specifically during the waves, you want to test them all. But there's no point in testing where the risk factors are minimal or the disease is very minimal. For example, people have been testing with these antigen kits, even with little bit of sore throat and all, which really doesn't make a huge difference, and then it puts additional load. Of course, I understand that with the 3 waves, now people have got used to it. But I think it is high time that as it's a community spread disease, we should come down to the same testing formula that we use for the H1N1 influenza epidemics that we have.
Okay, fine. So there is a, may I ask Professor Baijanti Madam to say something as being a chairperson? And there is a one question in there for you.
And also,
yes.
Dr. Sangram Singh is also there.
Yes.
So should I answer the question in the group that has been put out? Yeah.
Yes, well, that will be better. That will be better. But there is 2 questions. It will be better you see it and answer those questions further.
So the point 1 question is whether have I seen hyperglycemia in COVID infection? Yes, we saw a lot of hyperglycemia in COVID infection, and it is now well known because actually, if you look at the ACE2 inhibitor, ACE2 receptors, they are present in other organs also. That leads to even a pancreatic dysfunction, and many of the times the sugars jump up for nearly 4 to 6 months. Even patients who are, not diabetic, just impaired sugars, they all become over diabetic. But the patients, when followed up after 6 months, most of them tend to actually again come back to the baseline. Rarely, of course, we have seen hyperglycemia continuing. Maybe that is because it was an unmasking of the diabetes itself. Autoimmune diseases and COVID infections, yes, we have seen a lot of them. We have also published papers that includes TTPs, transverse myelitis. If we look at the acute point, we have also seen post-COVID arthritis, GB syndromes. We presented GB syndrome, and actually another important point is one of our patient had not only GB syndrome, he had SARS-CoV inside the CSF fluid. It was isolated from the CSF fluid. We also did if you look at the HLH itself as an autoimmune component, of course, that can be triggered by any infection. We did bone marrows in both first wave and second wave, and we saw a lot of changes which were mimicking the autoimmune picture. So SARS-CoV has done all these features, and it is very well known. Vaccine-induced hyperglycemia is much rarer as compared to the COVID-induced hyperglycemia.
Question.
May I ask
a
different
you,
question?
of course.
Thank you, sir,
No,
for a
sir,
very nice
I
presentation.
think last point, Dr. Harsh Sharma is saying, am I suggesting low COVID testing? I'm not suggesting low COVID testing. I'm suggesting pinpoint tests. There are two components. That's why I pointed out in my view. One is epidemiological perspective. If you think it is a new variant, immediately you want to restrict it, then you go ahead with the testing policy. Where you are having already a community spread, then you have to have pinpointed testing. Test those where you have to make a difference. That is why at the very beginning, I pointed out there is another perspective which you have to dip into, that is epidemiologist perspective. And whenever a new variant or a new virus comes in, where they want to restrict and decrease the amount of spread and look into it.
I think Dr. Sangram Singh wants to ask a question.
Thank you, sir, for such a nice presentation. So I have a query I want to ask you as a clinician. That what's your experience about the use of antiviral drugs during for those infected people with COVID-19? Should we use them or not? What's your experience as a
my experience, as well as what I would suggest is, antiviral drugs have a certain role in a very there is window in the clinical picture where you can use it. That is when the patient is actually high risk, who is just going into the moderate disease. There you actually have the antiviral role. Even in the remdesivir, in the Solidarity trial also, it was only the admission rates which were decreased. That means when you say that SpO2s, when they're falling below that 94 or suddenly coming down from 99 to 94, if you use it there, and within the first 7 to 10 days only, there's no point in going about using antivirals beyond that, where people have used randomly 14 days, 8 days, after 21st day again repeating it. All that is of no use. Now we are pretty clear about it. Use it in that group where they are actually going towards the moderate or severe. And once the patient lands up into that severe disease criteria as per the classification, that is below SpO2s of 90, constantly having a lot of lung injury, that is because of autoimmune. Then you don't see much benefit over there.
Thank you.
Dr. Vajantimala, please.
Yeah, thank you, Dr. Ujjala. I think Dr. Himanshu has given a very enlightening talk, very judicious. I think what he meant is the judicious testing, not low, not high, in community setup and in clinical setup. Thank you, Dr. Himanshu. I think we can move on to the next speaker. I invite Dr. Ujjala Ghosal, I think is the next speaker, who doesn't need any introduction in this forum and also in the microbiologist forum. She is a leader in our microbiology society and also an ambassador for North India and one of the leading lab for COVID-19 testing. So over to Dr. Ujjala.
So shall Dr. Vajantimala, shall I start?
Yes, Dr. Ujjala, please.
A very good morning to you all. Today's the topic of my presentation is issues with the diagnosis of emerging SARS-CoV-2 variants, a microbiologist's perspective. We all know that SARS-CoV-2 virus, which causes coronavirus disease 19, first emerged in Wuhan city in China nearly 2 years ago. It is an enveloped positive sense single-stranded RNA virus. And like other RNA viruses, it is prone to genetic evolution with the development of mutation over time, resulting in mutant variants. Since the reporting of the first case and the publication of first sequence in December 2019, the virus has undergone numerous mutations. And due to the continued evolution and their impact on public health, variants are reclassified as variants being monitored, variants of interest, and variants of concern. And this is showing you the genetic variation timeline of variants. So we have alpha variants, we have beta, then we had gamma, delta, and now we have Omicron. And this is again showing you the variants under circulation worldwide. That initially there was this original strain followed by alpha, then beta, and then followed by delta, which replaced all the strains and which in turn has now been replaced by Omicron. If you look at the Indian perspective, it is again picture is again similar. Initially original strain followed by others, then everything was replaced by delta, and then from January Omicron started, and then everything is now, most of the strains are now Omicron only. And since this is the strain in circulation in India and worldwide, most of our discussion will be towards this only. So regarding the mutation in Omicron, we all know that it carries more than 50 mutation, large number of mutation, which was not seen in combination before. And it includes more than 30 mutation on the spike protein itself. So this is showing the number of mutation in different variants, and you can see that Omicron has highest. This is showing you the effect of mutation, effect of variants on different and their effect on mutation in different variants. So alpha, you can see there is this deletion which leads to S gene dropout, that it doesn't amplify. Then because of this N501Y, there is increased affinity of the spike protein to ACE2 receptors, therefore enhancing the viral attachment and its subsequent entry into the host cell. Then similarly, we have beta and gamma. So beta, again, due to all these mutations, again, it increases the binding affinity for the ACE receptors. And gamma, again, due to all these mutation in receptor binding domain, there is increased affinity. In delta, there is no such thing. Whereas in Omicron, since it has large number of mutation, due to this deletion, there is S gene dropout. These mutations, they are predicted to have an overwhelming disruptive effect. And Omicron is more likely to have vaccine with 2 also. So let us see what is the impact of these variants on the diagnosis. The presence of mutation in the SARS-CoV-2 virus in a patient sample can potentially impact test performance. Molecular, antigen, and the serology test are affected by the viral mutation differently due to their inherent design differences of each test, as we'll discuss. So though there are many tests for the diagnosis of this virus, but the tests which are routinely used, more commonly used, these include the molecular tests like RT-PCR based tests. Then for the confirmation, we have sequencing like next generation sequencing or Sanger sequencing based for the S gene. Then for the rapid detection, we have antigen based tests. So regarding the RT-PCR, different targets are used. We use a structural protein like spike, envelope, and nucleocapsid.
we have genes. Then among the non-structural protein, we have RDRP, that is RNA-dependent RNA polymerase gene, replicase open reading frame. We also have hemagglutinin esterase and helicase genes and others. Then the antigen detection test is largely based on the detection of N antigen. So this picture is showing you with a basic overview. And most commonly screening gene target is E gene, and which is to be followed by the confirmatory genes, and the targets are RDRP or ORF or S gene or N gene and others too. The multiple targets are designed to detect more than one section of the SARS-CoV-2 genome to increase its sensitivity, which can otherwise be missed by if we use the single target. And this is showing you a normal amplification plot. This is very important that all controls should meet the standard requirement, and the positive results. You can see there's a sigmoid curve across the threshold line. This is the threshold line, and this is the sigmoid curve. And it should be within the 35 cycles for the target gene. So you can see here, this is showing the amplification plot for tag path. So this is you can see various targets like S gene, ORF, N gene, and MS2. What are the implications of variants on PCR? Most molecular tests for the SARS-CoV-2 are designed to detect specific RNA sequences found in the viral genome. Analysis of the FDA suggests that performance may be impacted, and the tests fall broadly in the two categories. First, the tests who are expected to fail to detect these variants, and second, the tests which are expected to detect the variants with a specific gene dropout detection pattern. Kits targeting a single gene can fail to detect the virus if it is mutated. For example, Meridian Biosciences Ribogene SARS-CoV-2 test fails to detect single target of N gene. Then we have another test, linear COVID-19 assay test, which targets the S gene and fails due to deletion at amino acid position 69 to 70 and mutation at 23599 to 23604 and a few other nucleotide position. So it may give false negative results, which can impact the outcome of these patients. Then we have tests which are expected to detect variant with a specific gene dropout detection pattern. So this S gene target failure, we get either weak signals or there may be complete failure of S gene target, which is caused by a deletion in this position of the gene. It was first seen in UK variant B1.1.7 lineage, which was a variant of concern, and also has been seen recently in the Omicron variant. So here is the amplification plot for the same, that you can see that though the other gene like N gene and MS2, they have amplified, but you can see that the S gene has not. So this S gene dropout or the S gene target failure may indicate, it can give you some clue that it may be a Omicron variant. And many tests like TagPath, combo kit, which targets many genes like N gene, S gene, MS2, and ORF, and many other kits detect multiple targets, may show S gene dropout pattern. SGTF, that is a S gene target failure, was observed in TagPath combo kit, and it is consistent with the certain mutation, include those seen in B1.1.7 variant. Using this approach, this variant has been detected at a faster rate than the previous surge of the infection. So the PCR is conducted using this kit. So if this is detected, then it is not you can think that it is unlikely to be Omicron, but if you could not detect this gene, it gives you some clue that, oh, it could be Omicron. Then it has to be confirmed by some other methods like sequence. Then there is something called N gene dropout too. So a 9 nucleotide deletion in the N gene spanning over this position results in an N gene dropout, and it is also known as S gene target failure. As you can see here, that though the internal control is fine, ORF has amplified, but here you can see that the N gene failed to amplify. So the presence of a gene dropout detection system pattern can signal that the sequencing should be considered to characterize this variant or confirm, and also for the confirmation. Pattern with one gene dropout may provide a signal that the Omicron variant will be present, but it is also important to note that around not all the patient samples with the Omicron variant display mutation. So the recent or the BA2 subvariant of the COVID is worldwide without a gene dropout detection pattern. Therefore, the Omicron variant may still be present without a gene dropout detection system. Despite this, the RT-PCR still remains the method of choice for the detection. Now, what about the antigen detection? Since most of the commercially available antigen tests, they target a viral nucleocapsid protein, that is the N gene, and the rapid antigen tests detect the presence of SARS-CoV-2 because most variants have S gene mutation. So it is targeting something else where the mutation is some. Antigen detection tests are generally less sensitive and are less likely to pick up very early infection as compared to the molecular test, as we all know. But these antigen tests, they do detect the Omicron variant. But the studies by FDA in collaboration with the NIH, the initial result shows that they may have reduced sensitivity for the Omicron variant. So that means they will be able to variant there, but their sensitivity may be low. But obviously these are initial results and need confirmation. Now, what is the role of genome sequencing in the diagnosis of SARS-CoV-2 variants? So we can identify, monitor, and assess the virus variants that may be more transmissible and are associated with increased disease severity. But the genome sequencing is not adequate for timely detection of the variants for public health responses. For example, the contact tracing and the calculation of prevalence of VOCs in a community. It is still, though there is a huge price drop, but it is still a relatively expensive method. It also requires huge investment in the equipment as well as training and bioinformatic analysis. For early detection and prevalence calculation of VOCs, alternative methods such as PCR-based assays that generate results in a few hours are obviously preferred. So we have many tests, including this endogenous new PCR kit called Omissure, which is approved by ICMR. This distinguishes the Omicron from Delta, wild type, and all other variants of concern. It targets 3 genes in a single tube and is fully multiplexed. It has a unique design. There is simultaneous detection of S gene target failure and S gene mutation amplification. And it can detect around 80% cases of Omicron. But 20% cases of still they may be missed, as this S gene dropout does not occur in around 20% cases. It is also important to note that S gene dropout may occur in some other variants also. So these are the few limitations, but at least will be able to detect around 80% cases within few hours with the help of this kit or other such kits. So now the take-home message: genetic variants will continue to rise regularly, which may lead to false negative results. Consider negative results in combination with the clinical observation, patient history, and epidemiological information. Consider repeat testing with a different molecular test, which has different genetic targets, if COVID-19 is still suspected. PCR test performance are impacted by certain variants due to mutation in different genes. So the test with multiple targets should be used for the detection. An alternative to genome sequences has to be chosen as it is time consuming, which defeats the purpose of timely diagnosis. Thank you.
Thank you, Dr. Ujjwala. It was a very nice presentation, very lucid and very excellent presentation. You have brought out certain facts very nicely, like the importance of RT-PCR in detecting the variants and its limitation, and how the sequencing will help us. It was a good, very nice message for how to detect the variants.
Thank you, Dr. Vajantiwala.
Thank you, thank you.
Thank you. Again, meet you during the, again, are welcome for the panel discussion.
Yeah,
right.
Thank you. I'll be there.
Please be there. Dr.
Thank you.
Gautam, are you there?
Yes. Yes, I am
Any
there.
question?
No, I don't have any, because it is such a nice presentation differentiating the different type of test which has done. People has got a confusion which test should be done, where the gene dropout is take place, what is the latest from ICMR on all this thing. So I think this should be published in a very wider margin platform, so that misunderstanding and we can generate more knowledge in this thing. That is the thing I just want. And nothing to say about you. You are the leader and you have done
Thank you so much.
so much
Thank you so much for
during
your
the
time.
COVID period, when nobody can think about it.
Thank you so much. So moving on to the next session, we would like to thank all the chairpersons for their kind comments and for being there with us. And now the next session is by Professor Uday Ghoshal. He will speak on short and long-term implication of COVID-19, and the chairpersons for this session are Professor Jayanti S. Sastry, Dr. Mini P. Singh, and Dr. Rajendra Singh. Dr. Jayanti S. Sastry, she is a professor and head TN Medical College and BYL Nair Hospital, and she is also the in charge of Molecular Diagnostic Referral Laboratory. Then Dr. Mini P. Singh, she's a professor in Department of Virology, PGIM Chandigarh, and Dr. Rajendra Singh is a senior principal scientist and professor in CDRI Lucknow. Now over to the chairpersons. Dr. Jayanti, please.
Good afternoon, everyone. Thank you, Dr. Ujjwala, for inviting me to chair this session. And it is indeed my great privilege and honor to introduce Dr. Uday Ghoshal, who's the professor and head of gastroenterology at SGPGI Lucknow. Of course, his work speaks volumes about him. He has about 336 publications. He has about 25 projects. He has received many awards. He has gold medals. He's a fellow of Rome Foundation, then USA, then fellow of the National Academy of Medical Science, fellow of Royal College of Physicians, Edinburgh, and Master of Indian Society of Gastroenterology, President of Indian Motility and Functional Diseases Association, Honorary Editor-in-Chief, Indian Journal of Gastroenterology, and editor and senior editors of a number of journal and advisory board member of Nature Reviews Gastroenterology and Hepatology. So today he is going to speak to us about COVID. And over to you, Dr. Uday. We are all looking forward to hearing you what COVID has how COVID has affected the gastrointestinal system, and what is your work in COVID with reference to gastrointestinal system. And we have some preliminary work with reference to microbiome, and I'm sure we will be in a position to share something about that after we listen to Dr. Uday. Over to you, Dr. Uday.
At the outset, Good morning. At the outset, I'd like to thank the Department of Microbiology, American Society of Microbiology, and the organizing team for giving me this opportunity to talk on short and long-term implication of COVID-19 on gastrointestinal tract. COVID-19 is caused by the severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. And SARS-CoV-2 enter into the cell through angiotensin converting enzyme 2 receptor. And angiotensin converting enzyme 2 receptor is not only present in the respiratory tract, but also in the gastrointestinal tract in plenty. Therefore, the virus also affects the gastrointestinal tract. The symptoms attributable to COVID-19 in the GI tract include anorexia, nausea, vomiting, diarrhea, abdominal pain, and discomfort. And as you can see here, almost 1 out of 5 patients with COVID-19 had GI tract symptom or gastrointestinal symptom in a meta-analysis of 60 studies, including 4,243 patients. This meta-analysis was done from Hong Kong. Also, the patient with COVID-19 may have gustatory and olfactory dysfunction. Taste dysfunction is not uncommon, and it occurs in 15 to 30% patients with COVID-19. Also, disturbance of the smell has been reported in a similar number of patients due to involvement of the olfactory nerve by the virus. And in fact, a study from India showed that actually 15% patients with COVID-19 had olfactory dysfunction. Now, what is the importance of GI tract symptom in patients with COVID-19? There are multiple studies now. This is one of the old meta-analysis published in Indian Journal of Gastroenterology in 2020, which showed that those patients who had gastrointestinal symptom actually had more severe illness, more chance of getting hospitalized, more chance of requiring ventilator, and dying more often. This is our data. When COVID-19 came to India in 2020, early part, actually Department of Microbiology must be complimented for quickly starting huge number of tests. And the initial month when they were doing the test, maybe during 1 to 1.5 month period, they did 16,065 samples. And these 16,065 samples gave a positive result in 252 patients. All these samples were not from SGPGI itself. A large chunk of this sample came through Integrated Disease Surveillance Program of India, which is a unique program to control infectious disease outbreak from the community. Now, what you can see here is of 252 patients, 208, that is almost 85%, were asymptomatic. And 44 were symptomatic. That means 15% patients with COVID-19 are symptomatic. 41% of these symptomatic patients had GI symptom, 34% had both GI and non-GI symptom, and GI symptom alone was present in 25%. That means 41% of these patients had a non-GI symptom. These are respiratory symptoms mainly. Whereas 34% had both GI and non-GI symptom, and 25%, note this figure, had only non-GI symptom. Only non-GI symptom, GI symptom. That means it is very important clinically that during an epidemic which is going on, we cannot just forget that patient with COVID-19 may present with GI symptom alone. What we found is those who had GI symptom, they more often had severe COVID-19, they more often had fatal outcome. What are the mechanisms of GI symptom? Let me recapitulate. Gut inflammation due to the immune activation in the GI tract by the virus. Structural damage including ulceration as shown in an occasional report by endoscopy and autopsy. Changes in intestinal permeability, direct effect of the virus on GI mucosa, increased mucosal serotonin, change in the gut microbiota, exaggerated GI motility, involvement of the nervous system of the GI tract, drugs used to treat this disease, and Clostridium difficile mediated. Rarely there have been reports of GI perforation and vascular thrombosis on the GI tract. This is a review that we are currently preparing, and there are multiple studies, mainly the case series and case report, which showed that there was perforation in the GI tract, even leading to fatal outcome, and this occurred due to hypoxia, maybe hypotension, endothelial injury, and microvascular thrombosis. Direct effect of the virus on the GI mucosa, increase IL-1, IL-6, TGF-beta, and increase in the von Willebrand factor. Now, can you get the virus RNA in feces of the patient with COVID-19? This is a meta-analysis which showed as far as 50% patient with COVID-19 may excrete the virus in the feces. More important than that is when there is a disappearance of the virus from the nasopharyngeal sample at about 1 to 2 weeks, the RNA in the virus persisted for long. Could it lead to enteric transmission or fecal-oral transmission of the virus? Yes, possible. But however, what we found is that the viral RNA load as reflected by sort of a fecal RNA cycle threshold is much lower, almost half of that in the nasopharynx. Hence, it is less likely that this will lead to a fecal-oral transmission. We all know that COVID-19 can have long-term respiratory consequences. Can it have similar long-term GI consequences? This is a review, or rather these are 2 reviews that we published long ago, in which we hypothesize that the virus, COVID-19 virus, may cause post-infection functional gastrointestinal disorder or disorder of gut-brain interaction in the long term. Why did we suspect that? This is because we all know COVID-19 can affect nerves, including enteric nerves. Angiotensin converting enzyme receptor is present in plenty in GI tract. During acute COVID-19, serotonin is increased in the GI mucosa, which is known to persist, can cause diarrhea, increase motility, and increase pain sensitivity. There are reports of increased intestinal permeability in patients with COVID-19. Immune activation and cell death in the GI tract is well known. Gut microbiota dysbiosis has been reported following COVID-19, and people have even shown that this gut microbiota dysbiosis can persist long. Fecal calprotectin has been shown to increase in patient with COVID-19. And fecal calprotectin is a marker of GI inflammation. Therefore, we can say GI inflammation does occur in patient with COVID-19. And central hypervigilance due to psychological distress and autonomic imbalance due to hypothalamic and brainstem inflammation can occur in COVID-19. All this suggests that there could be long-term GI consequences such as IBS following COVID-19. In fact, all these abnormalities listed in this review are also the abnormalities found in IBS patients. Now, what is post-infection IBS? When somebody develops IBS, which is a form of functional gastrointestinal disorder or disorder of gut-brain interaction, following an acute infectious illness characterized by fever, vomiting, diarrhea, and positive stool culture, without its presence earlier before this infection, is called post-infection IBS. And this is a meta-analysis done by Madhusudan Grover from Mayo Rochester, which showed almost 10 to 15% patients following any GI infection, such as acute gastroenteritis due to bacteria, protozoa, and virus, can be followed by post-infection IBS. Now, we hypothesized in 2020 that COVID-19 may lead to post-infection IBS. Now, we wanted to prove this concept. For this, we took 109 patients, mostly asymptomatic patients, diagnosed in the laboratory of microbiology. We followed them up telephonically at 1 month, 3 months, and 6 months. We took another 171 patients from Dhaka, Bangladesh, 3 hospitals. They inputted the data electronically. These are mostly symptomatic patients, severe patients requiring hospitalization. Similarly, we followed up. We also took the data of 264 healthy control, similarly followed up. And what we found that at 6 months, as you can see here, 15 patients developed IBS of about 280 patients. 5 patients developed IBS functional dyspepsia overlap, and 6 patients developed functional dyspepsia, which is a predominantly upper GI problem. IBS is predominantly a lower GI problem, and functional dyspepsia is predominantly upper GI problem. Most of the patients with IBS had diarrhea predominant subtype of IBS, and none of them had this problem or disease earlier before development of COVID-19. This is Kaplan-Meier curve. These are Kaplan-Meier curve comparing the cases and control. And as you can see, cases more often develop COVID post-infection IBS than control. What were the risk factors for its development? Presence of symptom during COVID-19, particularly GI symptom. In fact, those who had asymptomatic COVID-19 practically did not develop post-infection IBS or dyspepsia. Presence of anosmia and ageusia during the index illness, that is during the index COVID-19, comorbidity, and presence of symptom, GI symptom at 1 month and 3 months. That means those who had GI symptom at 1 month and 3 months, they were more likely to continue to be symptomatic at 6 months. Now, also we found that those who had GI symptom, they more often develop post-infection functional gastrointestinal disorder such as dyspepsia, IBS, and their overlap than those who did not have GI symptom. And what we found is those who were asymptomatic during initial COVID-19 practically did not develop post-infection functional gastrointestinal disorder. What could be the reason that those who had anosmia and ageusia during index illness develop post-infection functional gastrointestinal disorder? What does anosmia represent? Anosmia represents that the fellows olfactory nerves are involved. And the olfactory nerve, we all know, is a direct extension of the brain. And ageusia means that there is a taste sensation loss, and we all know taste is carried from the tongue by the glossopharyngeal nerve, which arises from medulla oblongata, and partly facial nerve, which arises from the pons. So therefore, loss of taste sensation means medulla oblongata is involved and pons is involved. And we all know the enteric nervous system, that is parasympathetic and sympathetic system of GI tract, actually is controlled by hypothalamus, and brain involvement means hypothalamic involvement. Brainstem involvement means the descending fibers which are coming from the hypothalamus are involved, facial nerve is involved, and glossopharyngeal nerve is involved. Therefore, we can easily explain why anosmia and ageusia were associated with the development of post-COVID FGID. Here, I'm very happy to tell you, this is the first study in the world on development of post-COVID-19 FGID. Now, psychological factors were important. Those who had psychological issues at 1 month and 3 months more often developed post-COVID-19 FGID. Now, the frequency of post-COVID-19 FGID in our study was about 9%, whereas we all know the bacterial gastroenteritis lead to post-infection IBS in about 15 to 20% patients. Similarly, protozoal infection lead to even higher frequency. And here I pulled the data of viral acute gastroenteritis. How often do they lead to post-COVID, post-viral FGID? And it is lower. Therefore, our frequency of 9% development is quite in accordance with the earlier studies with other viruses. I conclude that gastrointestinal manifestations are not uncommon among patients with COVID-19, and their presence is associated with severe disease and worse outcome. We found that symptomatic COVID-19, particularly with GI symptom, may develop post-COVID-19 FGID, and this is a consequence which has been reported first time in the world from India in collaboration with Bangladesh, and recently a paper has been accepted on this issue from US, which is the second study in the world. Thank you so much.
Thank you very much, Dr. Uday, for this excellent talk. I'm sure we should take it forward by looking at the microbiome, doing the cytokine analysis, and correlating all the symptoms with the various levels of cytokines. And congratulations for being the first to report this in the world. I pass it on to my co-chairs, Dr. Mini and Dr. Rajendra, to please take this discussion forward. Thank you very much.
Thank you, Dr. Jayanti, and I congratulate Dr. Uday for the excellent work done and the nice and lucid presentation, which has answered many of our queries. Sir, I have one question in this regards. Have you noticed any difference in presentation with regard to the GI symptoms in the various variants over a period of time? Like we have seen 3 waves with different variants. So is there any difference in regard to that also, whether these manifestations were more common with any particular variant?
Yeah, thank Dr. Mini for your nice question. It is a very important question. But let me tell you, I suspect the variants such as Omicron, which induces maybe less severe symptom, would lead to less of the GI symptoms, because GI symptom correlate with severity. But unfortunately, we did not compare the data in the second wave and the first wave versus third wave. So therefore, I don't know. We need more study on this. But your question is very thought-provoking, and maybe more studies will come up on this issue.
And sir, how long these symptoms last? Because I've heard many of my colleagues who have had this actually in the first wave. still complaining of functional GI disorders. And if there are any guidelines on the management of these disorders related to COVID, any specific therapy that you can suggest?
Yeah, what happens is your question again is very important. People have shown that the autonomic dysfunction that occur following COVID-19 can last up to 2 years. So when I say can last up to 2 years, doesn't mean that it disappear after 2 years, but people have documented even 2 years after having the disease, autonomic dysfunction persisted. Similarly, there are studies which showed that gut microbiota dysbiosis, like recently a study was published in Gastroenterology from Hong Kong. They showed gut microbiota dysbiosis lasted quite long, 6 to 9 months after development of the COVID-19. So what happens is in our study, we found that 6 months was the study point after which we analyzed the data, published the paper. But let me tell you, there are patients who has been now suffering for almost 1 year. But we have to learn from the model of post-infection IBS. So post-infection IBS study, there are 4, 2 from South Korea, 1 from China, and 1, I think, from Italy. And what these studies found is post-infection IBS is maybe a better IBS than the common garden variety of IBS. Post-infection IBS may be less permanent or less long-term than common garden variety of IBS. So I presume that these patients may have a better outcome, but we need to follow them up and learn from them.
Yeah. Thank you so much, sir. Thank you, sir. Over to Dr. Rajendra Singh, please.
Thank you, Dr. Jayanti and Dr. Meni. Thank you, Dr. Ghosal, for an excellent talk. He's our friend and collaborator, and I got an opportunity to work with him on some of these samples that he just talked about. And his research reminds me of a holistic approach. Say, for a disease like COVID-19, only pulmonologists and microbiologists cannot do the whole task, because it is not only a disease of the lungs. And so it goes much beyond that. And something like impact on gastrointestinal tract that may appear to be secondary in nature, but that can certainly affect the primary outcome in these patients. For example, like Dr. Himanshu talked, and it is now well known that the COVID-19 infection that is likely to result in hyperglycemia, even in otherwise non-diabetic patients. So unless we understand that, unless we understand the mechanism of that, we cannot really provide a holistic therapy to the patients. And we know now that microbiome that plays a significant role in the physiology of so many organs. Like we work regularly on fertility and infertility. So it is a distant organ from, gastrointestinal tract. But even there are studies that changes in gut microbiome, they affect fertility. So definitely studying these distant organs, otherwise distant organs that may appear to be, it is really very critical to understanding the pathophysiology of the disease, such as COVID-19, and then providing a holistic treatment. And regarding some of the interesting observations he made, I remember during the first wave, I think he was the first one to identify that the development of the gastrointestinal symptoms, that is a prominent predictor of the outcome of the disease. And I think they were also using an algorithm based on that to guide the treatment of the patients. So a very good talk, and I loved hearing you. I understood every single point in your presentation, and I'm sure your research will contribute to the patient care in COVID-19. Thank you.
Thank Dr. Rajendra. He is my friend and collaborator. And let me tell you, we did a study together. He did all the sequencing. Dr. Ujjwala contributed all the sample. And what we found is there is a relationship between mutation and the symptoms. So that I did not tell you because I think it still remains unpublished. But with his permission, let me tell you, there are certain mutant, particularly the later wave mutant like Omicron, is associated with a much less severe GI symptom than the earlier ones. But I'm taking his permission to tell this. This is an unpublished data. Now, second is that you asked me a very important question, how to treat this patient. Let me tell you, most of these patients of post-infection IBS following COVID-19 are diarrhea predominant IBS. Currently, there is no randomized controlled trial on this issue. Currently, there is no randomized controlled trial on this issue. Because there are studies to show that serotonin is increased in this patient. Serotonin is increased in the gut mucosa of this patient. So whenever I get a patient of post-COVID-19 with diarrhea, I treat them with serotonin blocker. There is no randomized controlled trial on this now, but I treat them with serotonin blocker. And serotonin blocker include ramosetron, ondansetron, and palinosetron. And my experience is the result is very good. Most patients actually get a good relief. Now, there are studies on diarrhea predominant IBS, not post-COVID IBS, which are diarrhea predominant IBS otherwise, multiple randomized controlled trial on ramosetron, ondansetron, and they showed they work in diarrheal IBS because diarrheal IBS is a high serotonin state and you are blocking the serotonin of gut mucosa, therefore you give them relief. But we need a randomized controlled trial to prove this issue scientifically that it works in the post-COVID-19 IBS. My experience is quite encouraging. Okay, thank you so much. If there is no other question, thank you again.
So thanks a lot. Thank you, Dr. Jayanti, Dr. Mini, Dr. Rajendra Singh, and thank you, Dr. Ghoshal. Now we move on to the next session, which is by Dr. Priya Abraham, and it is on mutation in SARS-CoV-2, effects of vaccine used in India. Now, the chairperson for this session are Dr. Amit Lahiri, Dr. Nivedita Gupta, and Dr. Rumi S.K. Marak. Dr. Amit Lahiri is a senior scientist in pharmacology in CDRI Lucknow. Dr. Nivedita Gupta is head of virology unit at the head office of ICMR in New Delhi, and Dr. Rumi S.K. Marak is a professor in the Department of Microbiology, SGPGIMS. Over to the chairpersons.
Hello.
Yes, please.
Yeah. So you can hear me, right, madam?
Yes, yes.
Yeah. So thank you. I'm happy to chair the session where Dr. Priya Abraham is going to give a lecture on mutation in COVID and effect in the vaccine. Dr. Abraham, as you can see, like she really needs no introduction. She's currently the director of NIV Pune. She has been working in cancer and viral diseases, and there are published seminal findings throughout the years. To talk about her research in COVID, we all know that she has been the first one, her group was isolated and characterized the SARS-CoV-2, and she has been in the forefront of COVID research, has collaborated with institute in India, globally, and various pharma company, which has ultimately given us the, the vaccine that we have been taking. So I would like to listen to her, and I will invite her to give her talk now. Thank you.
Good afternoon, everybody. I'm honored to be speaking at the CME on COVID-19, organized by SGPGI, the GADVASU, the IVS, and the ASM bodies that have organized this event. I would particularly like to also thank Dr. Ujala Ghoshal, who had invited me to give this lecture. I have been asked to speak about the impact of different virus variants on the vaccines that have been delivered in India. And let me now share my slides. You can see here depicted the 5 major variants of concern. And in India, we did also see variants of interest. We were fortunate in this institute to isolate the original Wuhan strain as early as March 2020, and thereon went on to isolate the Alpha variant. We were the first to isolate it in the entire world, and followed by the Beta, the Delta variants of concern, as well as the Zeta and Kappa variants of interest, as they were known then, and most recently, the variant of concern, Omicron. I'm proud to say, thanks to my colleagues led by Dr. Pragya Yadav and team, we have more than 330 different strains of the virus stashed away in our highest containment facility, the BSL-4 laboratory. We also were able to institute many screening antibody assays. We were the first to put out an indigenous whole virion-based ELISA. It was called the Kavach ELISA, which was used actually in the first sero survey and to screen individuals for clinical trials during the Covaxin studies and Covishield studies as well. We transferred it by an expression of interest to 7 companies. We also were able to standardize an in-house anti-spike RBD protein and nucleocapsid protein ELISAs, and also were very successful in setting up the microneutralization assay and plaque reduction utilization assays in this institute. These assays, particularly the MNT and the PRNTs, were very useful in studying the immunogenicity and protective efficacy of Syrian hamsters during our preclinical trials, in rhesus macaques, also in preclinical trials while we gave them after we gave them a virus challenge, and then also in phase 1, phase 2, and phase 3 clinical trials where we helped Bharat Biotech with this analysis, as well as studying the immune responses against the various variants of this virus. This is a picture of the PRNT, a laborious, cumbersome assay, but which has been extremely useful in studying the functional antibody response against various variants, and also in our preclinical and clinical studies for vaccine evaluation. So we come to the first variant of concern, the Alpha variant, and this on your left is the Wuhan strain, what we call the B.1 strain or the D614G strain, or even the G-clade strain. And this is the strain that was used to make Covaxin. So this is the prototype strain. This is the Alpha variant, and this is a variant belonging to the B.6 or the O-clade. And you can see here clearly the Alpha variant had no statistically significant difference in the GMT titers compared to the original prototype strain or the O-clade strain. When we come to the B.1.617, you may recall by late March, people began calling this variant as the double mutant because it had an E6484Q and an L452R amino acid mutation. When we compared that against the prototype strain and the Alpha variant, we found that neutralizing antibodies were lower with this strain. And when we looked at the what we call matched pair samples, we could see that comparing against a prototype strain, it was about 1.95-fold lower, and compared against the Alpha variant, it was about 1.8-fold lower here for neutralizing antibodies. When we looked at the natural infection versus vaccinees, we didn't see that difference simply because those that had natural infection were a mix of individuals that had been exposed to the original strain as well as some variants. So that difference did not come out with this particular variant and with many other variants, because natural infection is a mixed bag. And you know that this variant later went into 3 sublineages, B.1.617.1, the Kappa variant of interest, the B.1.617.2, the Delta variant and VOC, and the B.1.617.3 didn't really last out for too long. So we come to the Covaxin antibody response, again studying the Kappa variant, and these dots in green depict those who had vaccination versus those who had been infected, had COVID and then got vaccinated. These are red spots, and just vaccination is green spots. And when we looked at B.1 vaccinated versus infection, obviously those who were infected and then vaccinated, titers were higher, even for the Kappa variant, which is then known as a variant of interest. Today it's known as a variant under monitoring because it's hardly seen. You can see that there's an increase in antibody titers. But when we compared the prototype strain against the Kappa variant head to head, there was about a 1.94 drop in neutralizing antibody titers. And there was not a big difference when we looked at binding antibody against RBD versus those who were vaccinated and those who had recovered from infection and then got vaccinated, against depicting a mixed bag. When we compared the Beta variant and the Delta variant, which were the next most important VOC seen in our country, if you compare it against the prototype, you can see a number of other amino acid substitutions, both for the Beta and the Delta. And when we looked at Beta and Delta against a prototype strain, be it for vaccination or natural infection, we found the Beta was about 3-fold lower than the prototype, and the Delta was about 2.7-fold lower than the prototype. We also looked at the Zeta variant. It is again, it was then known as, it's also called the P2 variant. It was then known as a variant of interest, but now designated as a variant under monitoring. And when we looked at the antibody titers for spike RBD and nucleocapsid protein amongst natural infection and vaccinated, of course, for spike, it was a little higher because most of our vaccines are directed specifically at spike. But when we looked at those who had natural infection for nucleocapsid protein versus the vaccine, there was no stark difference. When we compared the natural infection and those who were vaccinated, the prototype versus this variant of interest then, now known as a variant under monitoring, the P2 or Zeta variant, we found that those who were vaccinated had a 1.92-fold drop in neutralizing antibody titers as compared to the prototype, and a 1.09 drop in titers, fold drop in titers as compared to the prototype as seen in natural infection. When we looked at recipients of Covishield, and this is an interesting study where we looked at individuals who had the first dose of vaccine, second dose of vaccine, those that had been previously infected and then had the first dose of vaccine, those who were previously infected and had the second dose of vaccine, those that had 2 full doses of vaccine and despite that had breakthrough infection, we could see across the board, be it for the prototype strain or for the Delta variant, antibody titers increased in those who had been previously exposed to the virus and then received either the first dose of vaccine or 2 doses of vaccine or even had a breakthrough infection. But of course, also across the board, you can see that the Delta GMTs were lower as compared to the Wuhan strain. We did studies even with the Sputnik vaccine, and the Sputnik vaccine too, in those that had been earlier exposed to the virus and then had 1 dose or 2 doses of vaccine, their GMTs were much higher as compared to those who had just had 1 dose of vaccine, 2 doses of vaccine versus those who had a combo of exposure to infection plus 1 or 2 doses of vaccine. You can see the titers are much higher here, and the fold differences obviously much less as compared to the prototype of B.1 strain because they had literally exposure, recovery, and then 1 or 2 rounds of vaccination. This too is published in the Journal of Travel Medicine is just been, just been accepted with the Journal of Travel Medicine and will be soon published. Many of you recall that in Uttar Pradesh, a small number of individuals had been accidentally given Covaxin after the first Covishield dose. And we studied the neutralizing antibody titers in these individuals. There are about 18 individuals. So we compared the homologous prime boost with Covaxin, with Covishield, and a heterologous prime boost, where individuals first got Covishield and then Covaxin. If you look at the actual titers against the y-axis, the heterologous prime boost of Covishield and Covaxin was higher, but it's much more stark here when you compare the neutralizing antibody titers against each of the variants of the virus, the prototype strain, the alpha VOC, the beta VOC, and the delta VOC. You can see here that the heterologous prime boost, Covishield plus Covaxin, was much higher as compared to the homologous prime boost across all the variants, as you can see here. This depicts data in individuals 6 months after they had 2 doses of Covaxin. This is binding antibody data, and this is neutralizing antibody data. This is using an ELISA. In fact, we used a CLIA, so we could actually get quantitative values. And these are, of course, the PRNT values. You can see after 6 months, these were the titers. And of course, with Omicron, it is the lowest neutralizing antibody titers as seen as compared to the beta, the delta, and the prototype strain. After the 3rd dose, or what we call the precautionary dose, was given here in our country, you can see that the GMTs of binding antibody as well as the neutralizing antibody was much, much higher. Of course, with Omicron being the lowest if you compare it across the other variants, but there was an increase in the antibody titers. And you can see pictorially the relative boosting of antibodies after the 3rd dose. We are here, this picture depicts after 2nd dose, after the 3rd dose, 2nd dose, 3rd dose, 2nd dose, 3rd dose, 2nd dose, 3rd dose across the 4 variants, the prototype, the delta, the beta, and Omicron. You can say uniformly across the board, there is an increase in antibody titers, and the fold differences are depicted in this slide, where for B.1, the prototype strain, after 2nd dose, there was a 19-fold increase after the 3rd dose. Likewise, for Delta, it was a 16-fold dose, Beta 14-fold dose, and Omicron 18-fold dose after the one got the 3rd dose after having got the primary vaccination of 2 doses. This is a ribbon model which depicts the ACE2 binding region and the RBD region of the spike protein here in green. And this shows the amino acid interactions between the ACE2 protein and the RBD region. This is the wild type version of the virus versus the Omicron strain. And you can see here that the pink circles here denote enhanced interaction. The blue circles denote poorer interaction. And what we are seeing here is, for instance, if you look at the T478K, the amino acid substitution, there's a net enhanced interaction. If we go to the N501Y, there is an increased interaction for Omicron. If we go to the Q493R, increased interaction. If you go to the the Q498R, also there is a net increased interaction between the RBD of the Omicron strain and the ACE2 protein. There were also some reduced interactions at K417N here, and the S477N shown here, as well as the G496S shown here. And if you look at the net interactions, there has been a net gain in higher interactions, which probably contributes to higher infectivity and higher transmissibility. When we compare the RBD region of the spike protein versus a candidate monoclonal antibody, this depicts particularly the IgG portion of the monoclonal antibody. Again, blue indicates poorer interaction, pink indicates higher interaction. If you look at this is the wild strain, this is the Omicron strain. If you look at this picture, clearly there's much more blue, suggesting that there is more loss of interaction than gain in interactions. As you can see here, the net gain, loss of interactions is more than the net gain of interactions. That probably explains the immune escape or the antibody escape. As you can see, for each of these amino acid substitutions, with Omicron, there is reduction in the amino acid interactions at each of these positions. So if you look at each of these amino acid positions, be it for transmissibility or for antibody escape or immune escape, almost in every position, Omicron is depicted almost at every position where both transmissibility and some amount of immune escape is also happening. And also it is seen with some of the other variants of interest. So if I were to summarize in terms of transmissibility, this is the grid from lowest to highest, gamma, beta, alpha, delta, and Omicron. In terms of antibody escape, alpha, gamma, delta, beta, and Omicron. And we have seen that pan out clinically, where the number of infections was so high in this third wave, mainly caused by Omicron. So to summarize, yes, we have seen differences in antibody responses to different variants. We see differences also across vaccines, the antibody responses provided by different vaccines. But among the vaccines that are approved and in use today, definitely there is some protection against the new variants. Currently, we can say that the vaccine definitely serves to protect against severe disease and fatal outcome. And we need to do everything possible to reduce the spread of this virus, because the more the virus spreads and infects more individuals, the more we run the risk of having more variants. And with that, thank you for your patient listening.
Okay. Dr. Nivedita, Dr. Rumi, are you there? Hello? Dr. Nivedita, Dr. Rumi, can you hear me?
Yes, I'm there. I'm there.
Please stay. Yes, please.
Yes, so thank you, Dr. Priya, for the excellent talk. I don't have any questions because I'm privy to most of the data and part of many studies that she has presented here. But one thing, as was rightly said at the end of her talk, that vaccines prevent against severe disease and hospitalization. Though the in vitro neutralization assays which are done in the laboratories are important indicators of the performance of the vaccines, but there has been raised a word of caution all across the world that such studies also need to be accompanied with real-time vaccine effectiveness data in the field. But then it also is limited by the epidemiological capacities available in countries. But along with in vitro neutralization assays, it's always important to have vaccine effectiveness data in real world, and also cell-mediated immunity data. Because whatever latest knowledge is emerging across the world, the CMI seems to be pretty well conserved across the board, though the neutralizing antibodies fluctuate. But overall, it is a very easy and important tool for determining the performance of vaccines in the backdrop of emerging variants. So thank you again for the excellent presentation, Dr. Priya. I would stop here.
Now the next presentation is by Dr. Pierre Coldin from Curie Institute, France. Dr. Rumi, may I like to take over? Dr. Amit, are you there?
Yes, yes. Okay. So, good morning, Pierre. Thank you, thank you for accepting us. So let us now hear from the research by Dr. Pierre Gregory Coulon. So Pierre has a PhD from France. Then he has moved to UCI for his first postdoctoral stint, where he has worked on various viruses and with immune response associated with them. Currently, he is in Curie Institute, Pasteur, in Dr. Benaros's lab, and he will talk on deciphering correlates of COVID-19 severity, exhausted CD4-CD8 T cells specific to conserved epitopes between SARS-CoV-2 and SARS-like coronavirus detected early on in severely ill patients. So let's hear Pierre's interesting presentation. Thank you.
That Dr. He is a very good friend and collaborator of Dr. Swam Prakash, and this is in continuation with the Dr. Prakash lecture. And this time the focus will be on human study looking at the T cell responses against conserved SARS-2 epitopes. Over to you.
Hello everyone. So I am Dr. Pierre Grégoire Coulon, and today I'm very pleased to be able to present some of the work I have done as a postdoc fellow in Dr. Ben Mohamed laboratory at UC Irvine in California with my colleague and friend Dr. Swam Prakash. First of all, I would like to thank the Sanjay Gandhi Postgraduate Institute of Medical Science and all the organizers who gave me the opportunity to present this work. So today, in continuation of Dr. Prakash's presentation, I will present you the human part of the work that we have done in the lab, where we try to find correlates of COVID-19 severity by characterizing the CD4 T cells and CD8 T cells immune response directed against conserved epitope between SARS-CoV-2 and SARS-like coronavirus that we included in our pan-coronavirus vaccine. And we did this by comparing the response, the T cell response in various COVID-19 patients, ranging from asymptomatic individual to severely ill patients who died from the disease. So since Dr. Prakash already introduced you on the project of the pan-coronavirus vaccine strategy that we have been working on in Dr. Ben Mohamed laboratory, I will be very brief in my introduction. The main point I want to highlight here is that most approved vaccines today, still today, are directed against the SARS coronavirus 2 only. So, and more specifically, those vaccines are aimed at producing neutralizing antibodies directed against the spike protein only, and more specifically against the first version of the spike protein, and not so much on inducing functional T cells. Unfortunately, we are all now witnessing the limitation of this kind of strategy with the emergence of antibodies escaping spike variants worldwide and the concomitant decline in efficacy of the major vaccines, even though those are still relatively effective in protecting against the most severe form of COVID-19. So today, only a handful of pharmaceutical companies and academic groups are developing universal pan-coronavirus vaccine, and there is no such vaccine currently. So we must find vaccine strategies that have high probability of working against potential new zoonotic coronaviruses. That's a first thing. And as we often say in the laboratory, the question is not so much if there will be a pandemic one day, a new pandemic, but when. So what are the main aim here? So as you will see in this slide, we went through 5 steps to identify, screen, and characterize our SARS-CoV-2 SARS-like coronavirus conserved and immunogenic epitope for our pan-coronavirus vaccine. So the first 3 steps were already presented by Dr. Prakash, so I'm not going to go through much detail here, but just briefly, we first did an in silico study to screen the CD4, the CD8, and the B-cell epitope that were highly conserved across SARS-CoV-2 variants, but also across SARS-like coronaviruses in human and in animal. Then we confirmed that those epitopes were immunogenic in vivo using HLA-A2-DR1 transgenic mice. And then, as Dr. Prakash showed you, we showed that peptides immunized SARS-CoV-2 challenged HLA-DR1, A2, and human ACE2 transgenic mice were at least partially protected, and we were able to prevent the most severe symptoms and the mortality of those mice. So now I will focus mainly on the human part of the work. So here we want to study the correlation between the observed immune response against those epitopes in various COVID-19 patients and the corresponding disease severity. So we want to see if more disease severity is associated with a lower immune response against our epitope or not. And then we want to characterize phenotypically and at the functional level those SARS-CoV-2 specific T cells in asymptomatic versus mild versus severely ill patient. So briefly again, here I'm showing you the conservancy of our 27 CD8 SARS-CoV-2 epitope and our 16 CD4 T-cell SARS-CoV-2 epitope. And you can see that the vast majority of them are sharing high similarity with epitope found in other human coronaviruses, such as human common cold coronaviruses, but also SARS or MERS. And they are also conserved, highly conserved, in various SARS-like coronaviruses, such as the one found in bats, pangolin, civet cat, and camel. So here is a summary of our pan-coronavirus multi-epitope vaccine with the 27 CD8 epitope and 16 CD4 epitopes that are highly conserved across various animal and human coronaviruses. And we also included a few B-cell epitopes in the spike that I'm not going to describe today. But focusing on the T-cells epitope, you can see that they are not only present in spike, but that they are focused on the whole SARS-CoV-2 proteome. So again, Dr. Prakash already showed you that triple transgenic mice expressing human AECE2 and human HLA, once after being immunized with those epitope peptide and then challenged with SARS-CoV-2, we are showing less symptoms and higher survivability compared to control, even when using SARS-CoV-2 variants when we did the infection. So in order to identify correlates of protection associated with the T-cell response against our epitope in COVID-19 patient, we collected blood sample from asymptomatic and symptomatic individuals, and then we extracted the PBMC from those blood sample and screened the genotype of those patients for HLA-A2 or DR1. Those cells were then stimulated for 5 days with the different CD8 and CD4 peptide individually, and then we performed different readouts such as interferon gamma ELISpot and FACS staining, looking at the functionality and the exhaustion, potential exhaustion of those cells in the various subgroup of patients. And then with the help of the clinician who assessed the severity score once the patient were discharged, so at the end of the hospital stay, so we were able to have 6 different category of patients, 0 from 5, with 0 being completely asymptomatic and 5 being the most severe, which are so the patient who died from COVID. And so basically you can think that 0, 1, and 2 are the patients with asymptomatic or mild disease, and 3, 4, and 5 are asymptomatic patients with severe disease that went to ICU. And we correlate those severity score with the T-cell response for each one of the epitopes. And here you have the gating strategy looking at the T-cell response, specific T-cell response, where we looked at the tetramer positive T-cells, so focusing on leukocyte, then CD3, CD8, CD4, and then the tetramer staining for against 5 of our CD8 epitope and 5 of our CD4 epitope. So here we are looking at interferon gamma ELISpot data. And we measured the CD4 T-cell responses following each individual epitope, HLA-DR1 epitope stimulation. And you can see when looking across the various severity group, from severity 5, patient who died in black, to severity 0, which are the asymptomatic patient in white, you can see that clearly for all the patients from category 5, 4, and 3 who developed severe disease, there was a very low T-cell response following each individual CD4 T-cell peptide stimulation. And inversely, if you look at the patients from category 0, 1, and 2 who developed either mild or no disease, you can see that the response were much higher. And this is representative data here from the ELISpot pictures, and you can see that severity 0 asymptomatic people were showing a really high T-cell response for almost all the various 16 epitopes, whereas looking at the mild case, the T-cell response was still quite good with a slight diminution, but then when we looked at the patient that developed the most severe disease, there was clearly a diminution in the number of spots. So this was true for almost all the epitopes except the ORF1AB, 5019, and 5033. and the OF7A98112. And you can see it here in this panel, where we are looking at the correlation between the severity and the number of spots. So the CD4 T cell response. So this is for all the CD4 T cells, all the CD4 ELU spots combined, and then for each individual epitopes. And you can see that for all of them, the slope is highly negative, with a R2 determination coefficient which is close to 1 and a high p-value. A p-value, a significative p-value, and excepted for those two epitope, this one here, which is non-significative, and the other one, which is right here, that shows also no significativity, and the slope, we can see it, which is close to 0. So here you can see the same type of interfungal spot data, but this time looking at the SARS-CoV-2 specific CD8 T cell response following the stimulation with each of the 27 individual HLA-A2 restricted peptide. And this is the same kind of conclusion, where the less severe patient, asymptomatic patient showing no symptoms, are showing a really high immune response, T cell immune response following stimulation by this spot. Whereas if you look at the patients who went to ICU with the most severe disease, the T cell response was really low. And again, this was true for the majority of the 27 epitopes, except for 5 of them, this one, this one, this one, this one, and this one. But all the other, you can see that there is a strong negative slope, R2 coefficient, which is close to 1, and a low p-value. So from those two first observations, we developed 3 main hypotheses in the lab. So the first one is that the symptomatic, severely ill patients could have severe lymphopenia. So for each individual cells, their function remained intact, but the number of and frequency of those specific T cells is too low. Or it could be also that the number and frequency of T cells broadly is too low, which has already been described in multiple studies. The second hypothesis is that in severely ill patients, those T cells could have shown impaired function in a process called T cell exhaustion. And finally, it could be both. So to answer the first hypothesis, meaning to see if the most severely ill patients were showing a sign of broad T-cell lymphopenia, we performed first a blood differential test looking at the total white blood cells number and the lymphocyte number. And as you can see, there is in the severely ill patient, there is a broad leukocytosis, so more white blood cells, but less T-cells, less lymphocyte. So then we wanted to know which type of lymphocyte was the most affected, and we looked by FACS staining. Here you can see that across the different severity group, this is mainly the CD3 T cells that are affected, and at the same level CD4 and CD8. So both CD4 and CD8 are affected at the same level. There is no difference in terms of the frequency of one compared to the other. It's not only the CD4, not only the CD8, it's both. So, okay, there is a broad T-cell lymphopenia in severely ill patients, as it has already been described multiple times. But what about the specific T cells? Are those CD4 specific T cells and CD8 specific T cells specific against our peptide specifically reduced compared to the whole broad T cell population? So, to answer that question, we looked at the tetramer positive T cells population after the staining with 5 tetramer targeting 5 HLA-DR1 restricted peptide and 5 tetramer targeting 5 HLA-A2 restricted epitopes. And you can see it clearly here, the patient from the asymptomatic group compared to the patient from the most severe group were showing in percentage way more tetramer positive T cells. So we found that not only there is a broad T-cell lymphopenia, but also that specific SARS-CoV-2 specific CD4 and CD8 T cells targeting our peptide were markedly reduced specifically in the most severely ill patients. Now we wanted to assess if those SARS-CoV-2 specific CD4 T cells were showing sign of exhaustion. And to do that, we looked by FACS at the expression of exhaustion markers such as PD-1 and TIGIT, and also the co-expression of CTLA-4 and TIM3. And here you can see, again, this is very impressive, that the patient from the asymptomatic group are showing almost no co-expression of those markers for all those 5 tetramer positive specific T cells. And same for CTLA-4 and TIM3. Whereas when looking at the most severely affected patients, those cells were highly co-expressing both CTLA-4 and TIM3 and TIGIT and PD-1 here. So then what we can conclude from that is that at the phenotypical level, at least, the specific SARS-CoV-2 specific CD4 T cells from the symptomatic patients, most symptomatic patients, seems to be exhausted. But to confirm that, we had to show that there was also a functional exhaustion on those cells, of those cells. And to do that, we looked at AIM marker for 1BB and OX40. And this time, in the asymptomatic patient group, you can see that they are co-expressing those two markers highly, compared to very much higher compared to the patient, the SARS-CoV-2 specific CD4 T cells coming from the symptomatic, very ill patient. So previously we gated on SARS-CoV-2 specific CD4 T cells, and here the gating is centered on the SARS-CoV-2 specific CD8 T cells this time, and basically this is the same type of result. So those T cells in severely ill patients are much more phenotypically exhausted compared to asymptomatic patients, and in asymptomatic patients those T cells are more functional. So obviously, in order to confirm that what we observed in our SARS-CoV-2 specific T cells in terms of exhaustion and reduction in frequency was characteristic of those T cells and not of all T cells, we looked at specific T cells from another pathogen, so targeting EBV BMFL1 protein. And you can see that when you compared across the different severity group the frequency of the EBV specific T cells, there is no difference. And same thing when you look at the exhaustion marker, the co-expression of PD-1, TIGIT, CTLA-4, TIM3 is not changed across the different severity group, nor the functional marker CD137 and CD134, where you can see that there is no difference again. So in conclusion, we have identified epitopes conserved in SARS-CoV-2 variants and SARS-like coronaviruses from human and animal. Those epitopes injected in transgenic mice were immunogenic and successfully attenuated the disease and symptoms and mortality after SARS-CoV-2 infection of those mice. Moreover, in COVID-19 patients, early on after the first symptoms onset, we were able to detect SARS-CoV-2 specific CD4 T cells and CD8 T cell response directed toward our conserved epitope. And the magnitude of this T cells response were negatively correlated with the disease severity, which means that they were positively correlated with better outcomes in patients. In COVID-19 asymptomatic patients, we found more functional interferon gamma positive specific CD4 and CD8 T cells. In COVID-19 asymptomatic patients, we found more T cells overall, but also more SARS-CoV-2 epitope-specific T cells especially. And finally, in severely ill patients, the SARS-CoV-2 specific CD4 and CD8 T cells directed toward our conserved epitope were showing phenotypical and functional exhaustion compared to asymptomatic patients. So one thing I have not had the time to develop here is that, if you remember well, those epitopes are conserved, right? And so there is a potential of cross-reactivity of those epitopes with, for example, human coronaviruses or other SARS-like coronaviruses. And one thing that we have found is that SARS-CoV-2 conserved epitopes that are the most correlated with less disease severity are the one for which we found the highest T cells, the higher T cell response in healthy non-exposed individuals. So the ones that are the most cross-reactive in healthy non-exposed individuals. So our hypothesis moving forward is that what we need to be able to develop less symptoms is a rapid establishment of a functional SARS-CoV-2 T cell response. Thus, previous cross-reactive T cell response could act as a pool of memory T cells capable of being recalled quickly, especially in asymptomatic and mild patients. Thank you for your attention. And I would like to thank particularly all my lab, specifically my PI, Dr. Al-Bashir Ben Mohamed, but also all my colleagues and friends who worked with me, Dr. Ruchi, Dr. Swayam, Dr. Nisha, and Dr. Soumya Brata and Dr. Awa. along with our collaborator, everyone in the animal facility who allows us to perform the mice work, but also everyone from the BSL-3 facility for the SARS-CoV-2 manipulation, and all the medical doctor from the UCI Medical Center. So again, thank you everyone, and I would be happy to answer any question you might have.
Thank you, Pierre, for the talk. So we are open to question if anyone has any question. So excellent presentation.
So I can't see any questions in the chat box. So I would like to take this opportunity to thank the speaker and also the chairperson. Thank you. What is this? Thank you for taking time out of a busy schedule. Thank you, Dr. Nivedita. Thank you, Amit. Thank you, PhD. Thank you, Dr. Rumi. And now the next presentation is by Dr. Yashpal Malik, and he will speak on prevention and control of SARS-CoV-2 One Health approach. Dr. Malik, please. And sorry, the chairperson, I would like to introduce the chairperson. The chairpersons for this session are Dr. Taposhi Ghosh, Dr. Rahul Dodhapkar, and Dr. Atul Garg. Dr. Taposhi Ghosh is a senior microbiologist and she is a professor in Calcutta School of Tropical Medicine and Hygiene. Dr. Rahul Dodhapkar is also professor in a department in virology, Jipper Puducherry, and Dr. Atul Garg, the professor, he is from our own department, he is in virology and is co-in charge of COVID-19 lab. So over to the chairpersons, please.
Thank you all. I'm Professor Taposhi Ghosh. Can you hear me?
Yes,
Hello?
very much. Welcome,
Okay.
welcome. We can hear you.
Okay. At the outset, I would like to thank the organizers for giving me the opportunity to be with you all. And I like to invite our next speaker, Professor Dr. Yashpal Singh Malik, for delivering his lecture. And it's a very pertinent today's. A brief introduction of Professor Yashpal Malik. He is actually the Dean of College of Animal Biotechnology, Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana. And he carried out his postdoctoral research in University of Minnesota, St. Paul, USA. He acquired advanced training in molecular virology from University of Minnesota, St. Paul, USA in 2005, Division of Virology, and then to the Institute of Ottawa and Wuhan Institute of Virology in China, first Dean of College of Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana. He also served as ICAR National Fellow at Premier Veterinary Institute of the country, Indian Veterinary Institute, IVRI, Ijat Nagar. Professor Malik is obtained his master's and PhD degree in microbiology from CCS Haryana Agricultural University, and he is also awarded many prestigious awards from Jawaharlal Nehru Awards from ICAR. Professor Malik has been editor-in-chief in Journal of Immunology, Immunopathology, and associate editor of Frontiers in Microbiology and Frontiers in Veterinary Sciences. Professor Malik is on advisory board of several professional academic societies, including Indian Virological Society, where he is serving as Secretary General and Secretary in World Society for Virology. So he needs actually no introduction. Over to Professor Yashpal Malik, please.
Professor Malik, are you there? The recorded lecture is there. Professor Malik. Yes, he had given a call 5 minutes ago. So we are again trying to contact. Maybe there are some issues. By the time I think there was a question for Dr. Ghoshal, he can, by the time he can answer. हम उनको बोल रहे हैं उनको क्वेश्चन करो. Hello. By the time there was a question for Dr. Ghoshal, you can please answer. I have sent on your WhatsApp.
It says, what is the personalized treatment for COVID-19?
One is that, and second, they are asking about the doses of ondansetron.
What was the?
क्या सर तो हमको तो दिखा रहा है सर का क्वेश्चन मैं बताऊं उधर दिखा उधर अच्छा बैठे.
Thank you for these two questions. The question number 2 I take first. That is, what is the doses of ondansetron? If anybody is treating a patient with IBS, be it post-COVID IBS, be it non-post-COVID IBS, remember ondansetron would be an off-label drug. Off-label means that this is not approved by Drug Controller General of India. So if it is not approved by Drug Controller General of India, you should prescribe carefully, because if a side effect happens today in the days of litigation, one can say this is not approved for this indication. But this study which was done on this was by Robin Spiller from Nottingham, UK. What he did is he used 2 to 4 milligram ondansetron, increasing to twice or thrice daily, increasing. So what you do is start with maybe 2 milligram twice daily, and then increase if the patient's stool frequency is not controlled and the form still remains liquid. So that is the dose. So there is no fixed dose. It is 2 milligram twice daily, increasing as you need. You can go up to 4 milligram thrice daily. This is the dose of ondansetron. Second question is, what is the personalized treatment of COVID-19? I tell you, this will be better said by an infectious disease specialist, because I'm a gastroenterologist. But let me tell you, very simple. Personalized treatment of COVID-19 is if a patient is asymptomatic, don't give him any antiviral, don't give him any drug, just let him be isolated, because he doesn't need treatment. Who needs treatment is people who can get infected from him. So what we have seen is a lot of patients who are mildly symptomatic or asymptomatic, particularly during the second wave, required used very aggressive therapies, and that led to a lot of problems. We have seen a diabetic patient getting steroid when he had asymptomatic COVID-19, and that led to his diabetes getting uncontrolled and he developing mucormycosis. So asymptomatic patient, no treatment, only isolation to prevent infection to others. Mildly symptomatic patient, only symptomatic therapy. For example, paracetamol, for example, maybe some people have used, like suppose antihistaminic, and whatever symptomatic therapy is needed, like running nose or blocked nose, people will give little bit of this thing, nasal drop like clotrimazole nasal drop, which is an sort of sympathomimetic, so that there is congestion getting reduced. There are some studies, very poor quality. They have even used betadine gargle or several other gargle to show that that may reduce the viral load. So these are simple measures, symptomatic measure. Otherwise, don't give anything. Don't give steroid. Don't give antiviral. Now, what about antiviral? If you look at all the studies, patient who had severe illness, critical illness, during the initial phase of illness, there has been mixed result on randomized controlled trial that antiviral, that is reducing the number of virus, might have done something good or better. Obviously, monitor their oxygen concentration, very, very important. And if they do a chest X-ray, if there is an oxygen concentration reduction chest X-ray, they must be under critical care, maybe require oxygen support, maybe requiring ventilator. Now, about steroid, there was a study from UK initially where they found severe COVID-19, particularly those who required ICU care. A randomized controlled trial of steroid led to marginally better outcome among patients with COVID-19, severe one, than those who were in placebo. But the steroid has been overused in the country. We know that during the second wave, and that led to bad effect rather than good effect. So therefore, be careful with the steroid. It is a double-edged sword. And the original study from UK had a very mixed result and must not be used in patient who has mild disease. Now, about chloroquine and all those ivermectin, the randomized controlled trial had mixed result. Randomized controlled trial had very mixed result. So question is mild disease, no role. And even initial studies, which are small sample size studies, showed a beneficial outcome, but subsequent large studies refuted those. So therefore, scientifically speaking, these drugs don't work. Thank you so much.
Professor Yashpal Singh is with us. His next talk is on the prevention and control of SARS-CoV-2 One Health approach. I welcome Professor Yashpal Singh Malik for his talk. Thank you.
Thank you very much. Am I audible to you?
Yes. Yes, sir.
So, let me share my slides. Is it visible to you now?
Yes.
First one.
Visible.
So, very good afternoon to one and all. All the distinguished speakers in past, and since morning we are discussing about coronavirus, specifically the COVID virus SARS-CoV-2. And one thing that has been necessitated in the past is regarding that we have to enter in One Health approach, because there are certain consequences which cannot be dealt alone. So something I will deal with, and a little bit will be on other viruses also where One Health is proving good. And the title is Prevention and Control of Emerging Viral Infections, and particularly the COVID, where a major emphasis has been paid on the One Health approach. If we go with the need of the R in past, influenza pandemic 1918, and almost 100 years later, we are also in the same situation. Nothing has changed, same mortality, infrastructure though has been improved, but still we are not able to control the infectious diseases. So what has been affecting the whole world is particularly three major areas, though there are little bit others also. One is the important is food and security. Food security and its safety. Then antimicrobial resistance. This is another grave area where another common platforms are there, where the VATs and the medicos, they are working together. Then emerging infectious disease. It is not leaving anybody in the world right now. If we are talking about, it started from one place and it has covered almost the global, all countries in the globe. So we have certain issues in the past. We cannot seek them as a separate issue. We have to take them all together. So all are interrelated. In the past, if we go just 2-3 decades only, we can see that list is increasing like anything. We have been in past affected by Ebola, then Zika. It is also in India, some places. Bird flu came in 1997 and it became a very permanent problem to the poultry industry. Then MERS came in certain parts of the world. H1N1, that is always a topic of discussion, that it is swine flu. Animals are playing certain important roles. So the number of viral infections specifically, but it is not limited to viruses. Other infections are also increasing these days because of the certain issues that I will just tell you in brief. So ultimately, what is said that viruses are becoming viral because everything, if the picture goes viral, every nomenclature we are using as a virus because it is fast spreading and it is more devastating than anything else. This is some of the epidemics and the global pandemics which affected. Even in the BC, we were having certain, but the time duration was very far. It was nearly 100 years or 200 years it was taking to come another disease. But if you see in the last 10 years, particularly the right side corner, it shows that within 10 years, we are having so many infectious diseases, which have been affecting the public, which have been affecting the even animal health. So number of infections are growing like anything, and there is a major discussion that what could be the reason behind this. Number of things have been identified like the winds, migratory birds, ocean current, role of the transport that has been seen even in the COVID when it appeared in 2019. Then the carriers which are taking our through the marine sea route. Then certain other things like land development and movement into the new area, rural to urban, slum areas, all are leading to the spread of the infectious organisms. And one good example, again, that is related with the movement of the livestock products, because this is one common area that animals are being used as the food animals. When there is a need of the food animal, they are transported throughout the world. And there are certain routes through which the pork, beef, or poultry that is transported throughout the world. So through that also there are all probabilities that the virus infection can spread. This is one common within almost 100 years. The time taken during the 1922 to reach one part to another was nearly 80 days. And it has been reduced to 8 hours because we have the fastest route. Going through the air, we are taking least time to go to the US. And along with us, some of other things can also move. Certain diseases, we have 3 stratas where diseases are there. One is typically the wild, another is the domestic animal, and then we are having the human population. So between these 3 strata, the chances of spillover is always there. Movement of diseases from wild to domestic and domestic to human cases. Even there are certain cases that from humans, again, the disease is going back to the animals. So reverse zoonosis is also possible. And what is the past literature shows that majority of the emerging infectious diseases are of zoonotic origin. Means something is hiding in the forest. Something is there within the animals. So it is the right time that we must collaborate, integrate our research approaches to find out that where is the niche, where is the ecological areas that we have to decipher the information. Same thing happened with the past with the bird flu H5N1, and now we have so many variants of this influenza H3N3, H3, H7, N8. So many variants are there, and they are moving somewhere in different species of the animals. H1N1 that appeared in the domestic fowl has gone to the wild aquatic birds, pigs, and ultimately reached to the humans in Hong Kong 1998. And since then, it is roaming around the world. In India, it entered in 2006. And similarly, the situation with H1N1, where there is a mixing of the viruses. Being segmented viruses, there are always chances that certain segments which are exchanging can lead to a generation of a new strain of the virus. So we don't have any conclusive idea that what could be the next. So we are always in probability, but we know that there are grave chances that whenever a virus evolves, it can lead to a dangerous situation. And many of the viruses are transboundary. There is no boundary-like thing in current world. Everything can cross the boundary, even the animal viruses, even the human viruses. So they are impacting the global at the global level. Whenever we talk about the viruses, the major culprit is the RNA viruses. If we go with the whatever has been done in the past, it shows that 44% of the emerging infectious diseases from the viruses are of the RNA viruses origin. And every year, whenever we are talking about the evolution of the new viruses, 2 to 3 viruses are of the RNA viruses. So they are higher probabilities to infect new host. More species spillover or the species jumping is there, and because they are having the shorter generation time. Rapid evolution rate, and they are more prone for the mutation changes. Mutation rate is much higher than any cellular host in case of the RNA viruses. If we compare it with the DNA virus, it is almost 5 times higher. So that's why this is always said that we must be very careful from the RNA virus evolution. We must emphasize on this group of the viruses. This is where we both meet, the medicos and the vets, they meet because we have a strong relationship with the animals. Because of some holy, because of the pets we are keeping at our homes, because of the need of the food with the increasing population, we have to shift or we have to depend ultimately because agricultural land is limited. So we have to go with the animal origin foods with the animal rich protein. These animals are traded also. They go from one country to another country. So there are chances of intermixing. So ultimately we are interconnected with the animals, and ultimately we are at a risk from animals. And similar way, I'm talking about the reverse zoonosis. So both ways we have to, we are at a one platform now, One Health. There are certain drivers which have been identified that definitely they are playing very important role. And almost all diseases, if you will see that Japanese encephalitis, this is linked with the irrigated rice production and pig farming. So ultimately the animal came and it acts as an amplifier host. So wherever there is a huge pig farming, intensive pig farming, there could be chances of that. Nipah is again linked with the intensification in the pig farming. This is one area in animal husbandry that people are shifting to the pig farming. This is very profitable. So that resulted in Nipah virus emergence in the Malaysia. If we talk about rabies, it is transmitted through the vampire bats to the cattle, and then ultimately to the humans also it is linked. So there are certain diseases which are linked with the deforestation, which are linked with the intensification in the livestock sector. So ultimately, again, this is one necessity that we have to talk together. something related to coronavirus, because this whole day, what we have spent as of now, it is revolving on coronaviruses. Coronaviruses are very old, though we start talking about that SARS came. But before SARS, 60 years back, there was an animal coronavirus that affected the poultry. And so many research have been done in the past in the animal coronaviruses. And ultimately, there also we have seen that viruses are changing, viruses are mutating in certain specific genes like S gene in animals also. There are evolution of a totally new virus in the coronavirus in animals. So there are certain lessons which are there to be learned from the animal coronaviruses. But if we talk about the human coronavirus, specifically the respiratory associated coronavirus that emerged in 2001, nearly SARS-CoV-1, and now in 2009, its new version SARS-CoV-2 that appeared. In between, there was another virus that was the MERS coronavirus. Again, there was involvement of the animal camels were there. So ultimately, when we go with the literature and when we analyze the data, it shows that speed is thrill, but it leads to the spread of bugs that kills. So ultimately, it has been linked with the SARS. A single person was found affected, and within 24 hours, it spread it to 5 countries. And within 30 days, it was reported in more than 24 countries, and leading to 8,000 cases and nearly 800 deaths. So this is the speed of spread of infectious diseases. It cannot be contained if we are not taking proper preventive measures. So speed is very important in case of the transmission in viruses. This is about Middle East respiratory syndrome virus, what I was talking about. That also spreaded very fast from the Middle East countries, affected from bats to camel and camel to human, and that affected again, and case fatality was very high, affected again 28 countries. And in some of the parts, still some of the MERS coronaviruses are isolated. This is the spread cycle of the virus SARS-CoV-2. It appeared somewhere in Wuhan, and within a month or 2 months, it became a challenging problem to the world, and it was declared by the WHO as a pandemic. But it originated from one place, and then it has covered almost the global population. So pathogens are under transition, and we are under continuous biorisk. This is the common question that always we talk about. If we go with the number of infections, the list is increasing. And on the top is right now we are discussing is the COVID, but Nipah is along with that. In India too, it is there in many parts of the country. Ebola, Zika, MERS-CoV-2, the list is very long. And in the counterpart, if we say, only the limited diseases are there, which we are able to either we are able to make them limited, restrict them, or we have successfully eradicated some, but only smallpox is the successful example. So there is a Pandora box. If we talk about any infection, the cases are increasing like anything. So what you cannot predict what is going to come, because what's going to come out of that, but you can predict that something is going to come. So on that we have to make certain preparedness programs. What is the main aim of the country right now, or the WHO and all the agencies, they are working that we have to prepare for the future, preventing from such pandemics. So this is what we got from a bat to the human population, a single case, and then the whole world. So number of SARS-CoV-2 or other coronaviruses are there, but if you go with this list, there are certain animals there are certain reservoirs which are playing very important role, including the bats, civet, pigs, camel, pangolin. So many are there, and some are there which still we are not able to conclude, or the researchable issues are going on. This is another important area that the World One Health is working on. There are nearly more than 50 infectious agents which are zoonotic, and rats are involved with that. So rat is one population that needs special attention if we want to control some of the infectious diseases. So ultimately, what looks to be the best possible way is that we have to come under one umbrella. If we want to go with the control of any disease, we have to go with the integrated disease surveillance program. We have to go with the preparedness and emergency response planning. And it can be done only through the government policies support. We have to enter with the multi-sectorial collaborations, and this all leads to the One Health platform. This is the simple one pic that shows that everything is common. All signatures are matching. If we think about the human thumb or the tree stump, everything matches. So signatures are matching and every problem is common. This is one start of that One Health era with the Rudolf Virchow, who first time identified that we must make one common saying that there is a common world. Human and animal medicine cannot be separated. So this was the start. So in the One Health, we have to make the coordination, communication, and collaborations very stronger. And there are certain areas where One Health is proving very good: food safety and security, the antimicrobial resistance, zoonosis, occupational health hazard, public health, climate change. So on this all, that One Health program is running successfully world over. This is one common that in nearly almost 40 years back, there were limited concepts on the One Health, with the publications nearly 1,000, and in 2021 it crossed more than 1 lakh. So that shows that impact that people are working on One Health approach, where vets started working for the human health, and still we are working, but it was nearly 1500 in the era 1500. Jacob Neuffer, he performed the cesarean first time, and then French veterinarian Jason Raman, he worked on the diphtheria, tetanus, toxide. So that is a very long or the old linkages with the human medicine. Even with the BCG vaccine, Kalmety is associated as a veterinary scientist. And even Doherty, who is well known as the immunologist, he is again a veterinarian and worked with the Zinkernagel and ultimately received the Nobel Prize. So there are certain areas where commonly we are working, including Dr. Barola, who is the vice president at Pfizer, and he was the one man associated with the COVID vaccine. So the government also realized, and there are certain common areas where ICR and ICMR, they are having collaborations, joint research projects, particularly influenza is one which has been identified. Some of the areas like rabies, it has been identified that commonly we will work. Then Department of Biotechnology also launched the One Health roadmap for India. So this is going on right now with the leading center at National Institute of Animal Health, Hyderabad. COVID testing is another good example of One Health. That number of veterinary labs in the country when the situation was very grave, and there was limited facilities for testing of the increasing number of the cases, veterinary department came forward, and ultimately so many laboratories were established. So this is the program, and these are certain veterinary universities which are still involved in the COVID testing in the country, including our institute at Gadwasu, from where I am. We are having the COVID testing laboratory and have tested nearly 6 lakh samples from the humans. And there are many other laboratories also at a high level which are working. These are certain publications that we published linking the animals and the human problems all together and covering the emergence of this COVID and relationship with the animals. What I was telling about that corona is not new for the animals. And there are changes in the coronavirus, particularly S gene. That is, again, we are talking about alpha, beta, gamma, delta, omicron, and so many on. Same thing happened in case of the animal coronavirus. There was one virus that is known as the transmissible gastroenteritis virus. This affects the pigs, and with the deletion of nearly 600 nucleotide in the S gene, the virus shifted its predilection. It became the respiratory virus in the pigs, from the enteric to the respiratory. So this is changing in almost everywhere. Corona is common to all. Same thing happened in case of the canine coronaviruses. Again, with the certain changes, a non-infecting or avirulent virus became the virulent virus. Some study we also performed to relate that how the coronaviruses of animals, they are matching with the SARS-CoV-2. And ultimately, we found that little bit they are divergent. They are not related genomically. Features are almost same. And the viruses which were isolated from the mink or the tiger or the pangolin, they are matching 99% with the human coronaviruses. Number of experiments have been done at the laboratory of Zhengli C in the China Wuhan Institute of Virology. They also confirmed that except certain species of the animals like ferret, cat, and dogs, rest of the animals are not having much susceptibility to the coronavirus too, like pigs, chicken, and ducks. There was a fear at a one time that don't eat the chicken, don't eat the ducks and pigs, because they can also carry the SARS-CoV-2. But it was clear that they are not susceptible to this virus. This is the pangolin story, and still we believe that pangolin played the very important role for the emergence of this SARS-CoV-2, because at the genetic level, there is much more matches with the virus, which was discovered. This is best example, I say, rabies, where all the One Health program is running best. So same way we have to go with other programs in the, like we are working on the Japanese encephalitis, we are working on the influenza. In the rabies, physician is involved with the vaccination and treat the victim. Veterinarians are vaccinating and sterilizing the dogs and cat. Wildlife experts are working with the oral vaccination. Ecologists are working with the sterilization and culling of the animals. Sanitarians are working, educators are working, and media is working to inform about the risk and prevention. So this is the best example of the One Health. This is another that because I am from the ICR and on deputation to the Gadwasu, at ICR number of good initiative taken under the One Health program. One of the bacteriologists, Dr. Alfred Lingard, he was the Britisher, and he established the first laboratory working on the animals in 1889 at Nainital Mukteswar. So that is again a good example. Now IVRI is working on number of zoonotic infections, working on glanders, working on buffalo pox, anthrax, so many areas where both human and animal, they are interrelated. So these are certain linkages with the wildlife, NCDC, then Nivedi, high security lab, what we are having in animal sciences. This is one at my institute where I was telling that we are working on the testing of the COVID at our center at Gadwasu, and tested more than 5.5 lakh samples as of now. We established it in 2020. 2020
yes,
it was
Dr.
established.
Malik, I'm just interrupting. Please, we are running short of time.
These are some of the collaborations which have been established between the veterinary and the medical institute world over, including the Royal Veterinary College and Sydney. So need of the hour is that we have to establish the collaboration, cooperation, and coordination. and we need commitment of the political and financial parties. So ultimately, we have to work on certain areas. We have to go with the skill development. We have to motivate. Technological driven attitude is needed right now. Many more viruses are being identified using the newer
Interrupt him. He
technologies.
is not listening to me.
So ultimately that this is the last slide, that we have to go with the One Health approach. It is not complex. It is a simple vision only, that everyone has to come together so that we can curb some of the important infection. So in the last, I end with the thank you very much to the organizers for organizing this event. Thank you very much, Dr. Ujala. Yes.
Yeah. Thank you, Dr. Yashpal, for a very nice talk. This is Dr. Rahul. And One Health is something which is very close to my heart as well. And this is something which is going to take us forward. And you have elucidated most of the points which need to be taken care when we are looking at One Health. It is not just veterinarians and medicals cooperating. We need a little broader group of environmentalists as well. But you have tried to elucidate most of the things. One thing which I would like to suggest beyond your talk is we need a mechanism, something like IDSP, which we have in medical diseases, where on a regular basis, whatever infections are seen in the community are documented and they are monitored by a central agency. A similar thing is required for animal diseases as well, at least the ones in the animal husbandry field, if not the wild, and collaboration of
Just
both.
to enrich before you shift to the second one, we have that established a way back. It is the National Institute of Epidemiology and Forecasting of the Disease. It is a big program in the country. We are working on all forecasting of the diseases and
So
epidemiology.
both IDSP and this need to work together so that
is a
animal
well-established
diseases can be
program in
forecasted
animal
in the
sciences.
humans.
This is not new for us. This is a very old program that we are running. This is the time to come together only.
Yeah, that's what I'm saying.
Because
If it is
people
there,
are not knowing that
they
at
need
what
to work
level
together.
we are working. We are working at a village level to a person who is
the best.
keeping a goat or a cattle. We have that information with us, that how many infections are there in one village. So that needs to be then matched
Collaborated with the human disease system, and together they need to take a call if there is something in a particular area.
Yes,
the humans in that area need to be screened if at all there is anything which needs to be seen in the humans, so that we can see the transmissibility.
Exactly.
Very nice talk, sir.
Exactly. Thank you very much, Dr. Rahul.
Thank you.
Thank you, Dr. Ghosh. Dr. Ujala is there or left?
She's there.
Yes, very much.
Oh,
Thank you, Dr.
hello.
Yashpal.
I was not able to see you. Oh, okay, great. Good to see you.
Thank you, Dr. Yashpal, for a very lucid presentation. I would also like to take this opportunity to thank all the chairperson, Dr. Tapushi Ghosh, Dr. Rahul, and Dr. Atul Gar. Thank you all. Now moving over to the last session, that is a panel discussion on coronavirus disease in India, a success story. And I would like to welcome all the panelists. Baijanti Mala Mishra, Dr. She has already been introduced. Then Director IDSP, Infectious Disease Surveillance Program, Dr. Vikasendhu Agarwal, Nodal Officer of our Rajdhani COVID Hospital and Head Emergency Medicine, Professor R.K. Singh, and Dr. Atul Gar from our Associate Professor from our department. Are you all there? Dr. Singh, have you joined?
Yes, ma'am, I have joined.
Welcome. Dr. Baijanti Mala?
Yes. Yeah, hello, Dr. Ujjwala. Good afternoon. I'm here.
Okay, welcome to this panel. So, should we start?
Yeah, sure.
So, this is the last session for the day. So, at the outset, I would like to ask Dr. Singh that we have seen that in many diseases, it so happens that it affects a particular system, organ system. But in contrast, the SARS-CoV-2 or the SARS coronavirus, it affects a large number of systems and has very wide range of health impacts. So why so? What do you think?
This is primarily because it binds to the ACE receptors, and wherever you have ACE receptors in the body, those organs are very likely to get involved. And ACE receptors are prevalent in multiple sites in the body, and therefore this pattern you are observing. Second is also because of the immune capacity of an individual also decides if I'm get stuck with an ACE receptor, whether I'll bounce back of the illness, I will be with the illness, or I will die with the illness. These all depends on my immune structure, as well as the amount of load I will imbibe when I'm get infected. So multiple organs get infected, yes, primarily the respiratory, but other systems are also known to be involved with the COVID.
Yeah. So thank you very much. So as he rightly told that initially we thought it is only primarily the respiratory system, but now as the time passed by, we know that it's a number of systems. Like you had listened to Dr. Ghoshal that it has many other systems involved, other respiratory system. So and sometimes it may present with the. It is not that all the time you will have respiratory system. Sometimes it may present only with the GI symptoms. So we have to take care of that. So thank you, Dr. Singh. Now my next question is to Dr. Vajantimala. So as the spectrum of disease has changed, like initially during the second wave or so, we saw such severe disease, and now progressively we see milder cases. So what should be our approach for diagnosis? Right, initially, like we were testing huge number of sample. We tested 10,000, 12,000. Now it has decreased, but even now we are testing 3,000, 4,000 samples. So what do you think as a microbiologist, what should be the correct approach for the diagnosis?
Yeah, initially actually we didn't, nobody knew about the virus. Now within these 2 years, we to some extent or to a large extent, we have understood how the virus behaves and what is the severity or how the variant comes, or how maybe about the variants, and we have certain knowledge or certain understanding about the virus. So at this stage, particularly after the Omicron wave, or who should be tested if you ask, it should always be decided at this stage as per the community transmission. What is the level of community transmission? And now we know that particularly now in the month of March or after the second half of February, we are now going through a very low level of community transmission. So at this stage, the testing should be very much focused where exactly it is required. Like already the ICMR has changed its guideline who should be tested. Like previously we used to do all the pre-op cases and a lot of community, a lot of asymptomatic individuals. So all those have already been stopped by ICMR. But if situation arises, because this is a evolving things. So as we will evolve through the process, as per the situation of COVID, this guideline will keep on changing.
Yeah, thank you very much, Dr. Vajantimala. So you talked of community transmission. So my next question will be to Dr. Vikas Sindhu, who, as I told, is a director IDSP who is looking after all this COVID-19. And we are doing so many cases. So you listen that we are doing so much cases. So he's the person behind, and he is like various samples, how they are transported to the lab. He's taking care of all this and many more things. So I would like to ask him that since as the COVID cases are largely decreasing, can we now consider that it is endemic in our country?
Good afternoon, Dr.
Good afternoon.
First of all, I will like to inform you that there was a whole team which was working for sampling and testing and laboratory strengthening led by Dr. Harsh Sharma, who has now retired, but he was the key person who was doing it all. I was also supporting him. And secondly, the question to which you are asking an answer for, whether we can consider it as an endemic situation as of now. You know, there are certain stages in the evolution of a pandemic, like when the global risk is too high, you declare it as a pandemic, then there is a stage of deceleration and then you say there is a stage of control or pandemic stage. So as of now, if you look at the global data as late as on March 14th, the number of new cases reported was more than 12 million. So the numbers are nowhere going down to the stage where we can call it endemic. Yes, the numbers are decreasing, but then there are spurts, and the spread is too far and too wide, and many countries are reporting simultaneously increase in number of cases as of the last week of March itself. So I don't think we can call it a pandemic situation and an endemic situation as of now. It is still a raging pandemic, and we have to wait for some more time before we declare it as endemic. That is my opinion, and that is the WHO opinion as well.
Okay, thank you very much. And as you rightly pointed out, Dr. Harsh was mainly responsible for this and his team. So I would also like to take this opportunity to thank him and the whole team. And in morning hours, he was there. I don't know now he's with us or not, but again, thanks are due to him and the whole team. So my next question is to Dr. Atul Garg. Since we have seen a lot of changes and now the cases have, since you are with us since the beginning and you are in charge of the lab also. So can you please tell us, should be any change in the strategy for the diagnosis? Like from the beginning, we are concentrating on molecular method or the RT-PCR. So now should we move from this to detection of antibodies, as has been done in many Western countries? They are focusing more on detection of antibodies, whereas we are focusing more on RT-PCR. So is there a time to switch on? What is your opinion?
Ma'am, in my opinion, I'll not like to recommend to move to antibody testing for diagnosis of acute cases of COVID-19, because antibody positivity can be because of a natural infection. It can be because of vaccination. In our country, we have already inoculated more than 180 crore doses. So if we get antibody positivity, we will never know whether it is because of the natural infection or of the vaccination. And like in hepatitis B, there is a cutoff that, okay, antibodies against this level are protective. There is no such quantitative cutoffs in COVID. And in fact, the FDA in July last year, they reported that antibodies can be used for the surveillance, but they should not be used for acute diagnosis. For acute diagnosis, the focus is mainly on either real-time PCR or antigen detection. Many countries like Japan, Finland, they say that antigen detection test using chemiluminescence is equivalent to RT-PCR. It can be accepted. So antibody cannot replace the antigen or real-time PCR testing currently.
But isn't the sensitivity of, you talked of antigen detection test, isn't the sensitivity of antigen detection test is too low as compared to RT-PCR?
Yes, ma'am, I agree with you. That is why the countries which in their passport which are relying on antigen detection, they are talking of antigen detection using chemiluminescence assay. Like antigen detection kits are of two types. One is the rapid antigen kits. These are lateral flow assay, and second is a chemiluminescence based assay. Chemiluminescence, there are several publications on this CLEA technology. They have a high sensitivity and specificity when they were compared with the gold standard test.
Okay, thank you, Dr. Atul. My next question is to Dr. R.K. Singh. We have seen that in different waves, the severity of disease was different. So, the second wave was much more severe. Then we had seen very mild cases, and during this third wave. So, what do you feel that is it due to vaccination, or there is this variant? As they say, this different variant have they?
I think if we compare, yeah, ma'am. I think if we compare the wild strain from the Alpha or from the Delta and the Omicron, there is and in between there is vaccination, the exposure levels are also going up. So there is natural immunity as well as vaccine-induced immunity that has crept up. And therefore, this could be one of the reasons why the intensity of the infection or the has reduced over time. So what we saw in the alpha, it was very different. What we saw in the delta, we were really overwhelmed, and the intensity was also, vaccination had yet not crept in during the delta. It's only in the latter end of the delta and in these before the start of the Omicron that vaccination really picked up. And the strain, therefore the strains that have come out have been now hit by two aspects. One, your natural infection raised immunity. Second is the vaccine induced immunity. And probably therefore you are finding the intensity of the infections is reducing over time. I do not know what the next strain will come. Well, but whatever comes, it has to be so dominant, so strong, because it has to now bypass our natural immunity of the COVID. It has to bypass our vaccine-induced immunity as well. So it will be very, very strong. Otherwise, it will have no influence on us over time. That is what I feel. So both, yes, natural as well as vaccine is both of them are playing roles in the intensity with which we are being hit by this COVID strains. Both of them are playing.
Okay. Thank you. So since you talked of variants, so my next question will be to Dr. Vikas Sendhu. And I would like to know like the role of, because genome sequencing, we had a very limited capacity. And so what is and it also like very, as we all know, very hugely costly, and then it takes time, then you have to have the capacity building also, so many issues. So in your opinion, what is the role of genomic sequencing for the diagnosis of variants and also for the tracing, because contact tracing is also important for tracing of this contact tracing and for the diagnosis also. How far we can rely on this, or should we go for some other methods since we are doing huge number of testing?
You know, genome sequencing, though it is a time consuming and costly affair and needs a lot of expertise, but it is mandatory to keep ourselves updated about the evolving situation. And our capacity was limited in the beginning, but then we did develop a few labs in UP like ICMR, NBRI, BHU, now KGMU is also doing. And Government of India has a centralized system through INSACOG where they can shift samples from one place to another. We send samples to the nearest laboratory, and we have been able to do quite a big number of genome sequences. And sometimes in the administrative circles, they ask the question, what is the need of genome sequencing when the basic treatment remains the same? You know, when you get to know about an evolving variant, and you know there is some significant difference in the genetic structure of a new variant anywhere globally, then you prepare. So that is the most important aspect about genome sequencing, that whenever there is a new genome emerging anywhere across the globe, you start preparing for another upsurge in number of cases, another uptick in number of districts which will be affected, and you start doing your mock drills, your full rehearsals. You fill the gaps wherever they are in your hospital preparedness. So genome sequencing, I think it must continue. As far as contacts are concerned, we do genome sequencing based on some definite criteria which are laid down by Government of India. And contact tracing is an entirely different vertical, that is for knowing the spread of a particular variant, how fast it is spreading, how much it is affecting, what is the R0. So that all is derived through contact tracing and the number of actual cases, new cases. But genome sequencing, yes, it is useful. It keeps us prepared. It keeps us informed. And we must continue with some baseline genome sequencing so that we are not caught off guard.
Yes, thank you. So I agree with you when you say it is important, but I think our objective should be clear, that what do you want? What is our aim? And yes, accordingly, we should use it wisely.
Yes,
whenever
yes,
indicated.
Yes,
Right?
we cannot go for very large numbers, but some
Yes,
certain numbers, some certain percent.
Yeah.
Yes.
Yeah. So we should know what we are looking for. Thank you. So my next question is to Dr. R.K. Singh. I just wanted to know that we talk of a lot of antiviral treatment or steroid and other such things. So when these are really indicated, obviously now we have such many more mild cases. So what is the, now in the current scenario, what is the role of antiviral treatment or the glucocorticoids and other such therapies for COVID-19 patients?
Asymptomatic patients practically don't require anything. Milder cases, immunocompetent patients, they don't require anything. But milder symptoms in patients who are immunocompromised, already on immunosuppressants, there may be a role of some of the emergency use authorization drugs are there. Some of them can come in if you are immunosuppressed, post-renal transplant on drugs, and then there is a risk of having this and you're just having mild symptoms or you're exposed. So there is a small caveat of emergency use authorization of monoclonal antibodies in that segment, that too in the first 72 hours of the origin or the exposure period. So there is some role. Again, for Omicron, this doesn't apply. Why? Because these monoclonal antibodies, except sotrovir, Vmab, none is recommended for Omicron variants. Okay. Now, as the cascade of the illness rises, say you go into a severe element of the illness, or there is requirement of low flow oxygen, high flow oxygen, or non-invasive ventilation or mechanical ventilation, there the cascade breaks up into 3 parts. So for low flow oxygen, so dexamethasone comes in all these 3 elements. So the dose being 6 milligram orally or IV once a day for 10 days. So this is the cascade for low flow, high flow, NIV, mechanical ventilation or ECMO. Then the most controversial during the COVID has been remdesivir. Now for the low flow, still there is recommendation for remdesivir. But for high flow or NIV patients with or without remdesivir, there is a caveat there. You may give it, you may not give it. There is a caveat. But for patients who are mechanically ventilated on ECMO, you may not have the best results with remdesivir in that group. Then in the low flow oxygen, if you are immunocompromised, you can evaluate for the component which I just now told you, monoclonal antibodies, if there is a risk. And if you are for the high flow oxygen or the NIV ventilation, you are there. And if you are within the first 2 days of onset within the ICU, you can add on tocilizumab or baricitinib in this group. Once you get mechanically ventilated or on an ECMO, and you are deteriorating in the first 24-48 hours of within this hospital ICU admission, then you can add on tocilizumab. So these are the elements where this therapy is going. If there is no oxygen, there is no steroid, just to remember. No oxygen, no steroids, okay? As the oxygen comes and the intensity of this oxygen increases, then the elements of additional therapies like tocilizumab, baricitinib, or newer medications in this group are coming up. Dose can be escalated. See, this is how you have to approach these patients. And baricitinib, again, we have tried it on some of our patients here, patients who were having high risk of going on to mechanical ventilation, but the results, I can't quote on the results. Why? Because the number of cases were very few, but it is an immunosuppressant. So you have to be careful when to choose this medication. Not in the extreme cases. Earlier the treatment started with an antiviral, the better the results. As the cascade goes on, all these medications really don't help you when you are really sicker. So more life support therapies come on there. As far as other medications are also there, like ivermectin, the US or the other data does not support it, but the UP guidelines and the R guidelines, we do support the idea that it may have a role. There are both plus and minus sides of the story. So maybe it is still evolving, I would say. And I think this is most common things that we have used for the COVID in our cases.
Okay, thank you. But if you remember, we had a lot of cases of mucormycosis. Though we had like mortality in India was very less, even less 1% to the tune of 1% of. So as contrast to the Western countries where the mortality rate was very high, but they had very few cases of mucormycosis in contrast to us. So what, in your opinion,
I think
what could be the cause? Any of us feel that is it misuse of glucocorticosteroids or
we are the capital of diabetes, we are the capital of mucormycosis, we are the capital of sicker hospitals. So the hospitals themselves are sick, the quality index is not there. So when you combine all of them with steroids, uncontrolled steroid, you will have mucormycosis. Okay, so this is the cascade, I would say. So at our center, we were receiving a lot of patients referred from other places. We did not have mucormycosis as a primary case in our center. So because one of the reasons were maybe that the quality of care was better, the control was better, the steroids were used more judiciously, and we were very upfront in using the antifungal. That is also, I can add, we had immunosuppressed patients. We did not wait too much. So an empirical regime did come in for diabetic patients, uncontrolled patients, 3 or 4 days with broad spectrum antibiotics, inflammatory markers not improving, changing over. So we were upfront in using some of these, and therefore our center, I can say, which is the apex center for COVID in this state of UP, we did not have primary mucormycosis at our places. They were all secondaries coming from other centers referred to us.
Okay.
This much.
Okay. Thank you very much. So since a large proportion of our population is already vaccinated, so my next question to Dr. Atul is that how long the immunity lasts? This question comes frequently to our mind that we have taken, we have taken both the doses of vaccine. And now we talk of different variants which come from time to time. So when the next should be, is there a need of third dose, or when how long this will provide protection to us?
In this regard, I'll like to say that by vaccination taking 2 doses, there are a lot of published data in the literature, which says that majority of patients have good immune response for first 3 months, and between 3 to 6 months, it slowly wanes off depending from person to person. But majority of these studies are based on detection of IgG antibodies against spike proteins using chemiluminescence assay. So whether these antibodies are protective or non-protective, that is whether they're neutralizing or non-neutralizing, this we cannot say. Second, we don't have any cutoffs. Like if we had a cutoff, then we can say yes, for first 4 months we have the antibodies are above the cutoff protective levels. Since there are no cutoffs, we cannot say whether the antibodies which were there, good antibodies for the first 3 or 4 months, they were protective or not. And again, there is a lot of literature which says that the earlier antibodies which were there, even the patients who had an infection with Delta waves and 2 doses of vaccines, the antibodies had a very low protection against Omicrons. So these coming of the new variants, this will always cause a problem. But theoretically, yes, we have the antibodies for 3 to 6 months.
Okay. On a similar note, my next question is to Dr. Vaijayanti Mala. Since different countries have different time duration between the doses or the boosters, so what should, in your opinion, what should be the ideal time between the 2 doses, and should it be it should be homologous or heterologous, and different types, since this different type of vaccine, they impact the duration. Do the different type of vaccine, they impact the duration between the doses, or what is your opinion?
In general, if we see the dose between the first prime and first and second, it is like 3 to 4 weeks in different countries. Between the second dose and booster, it is like 3 to 6 months in different countries. And this is usually like taken on the basis of, as Dr. Atul mentioned, like most of the studies, what it has shown that the effective immunity or the neutralizing antibody, it persists for 3 months. So that must be the reason for that gap of booster from second dose to booster. Even in India, we have 3 months gap, and in many countries it is 4 to 6 months. WHO also says it is 4 to 6 months. Now, regarding this, I think this is about the interval of first, second, and between the second and the booster. This is the basis of taking or determining this gap.
Okay, thank you. And regarding this, whether we should receive the homologous dose or the heterologous?
Yeah, yes. Homologous, actually, many countries initially it came up with the homologous booster. But now that we are getting evidences for this immune response, better immune response with heterologous. Even Dr. Priya also showed that heterologous is showing better, and WHO also now accepts that mix match combination is accepted by WHO. So I think with the guideline also, we are going to get the heterologous combination is as an acceptable mode. And now all the recent evidences, if you see the studies published in Lancet or recently March, NEJM, all studies are showing heterologous is showing better response.
Okay, right. Okay, thank you. And I just wanted to know from Dr. Singh that is there any way, of course, diagnosis is important, we all are doing, but clinically because we are doing testing a lot. So is there any way for you to understand or to differ it from, like, you can, whether clinically can we say that or differentiate or have a clue to differentiate
clinically the most differentiation, if I'm not wrong, you are asking between influenza versus the COVID. Now that is the most, now clinically there is no way in which you be able to differentiate. The correct answer is there is no way except you do a testing. That is the short answer to this. There is no way of clinically knowing whether you have influenza versus a COVID.
Okay, so even after seeing so many cases and having
No.
so much experience past 2 years,
No.
it is not possible.
If there
for
is,
us to
what I will do is, if there is a pandemic going on, then my suspicions will be more towards that area. Otherwise, if in a low, say in an endemic area, there is hardly any way where my suspicion is also high and I can differentiate in my sitting in my OPD between an influenza and a COVID. It will be difficult for me. I need the testing.
Okay, and my last question to Dr. Vikas Sindhu is, what is it, since this third wave is almost going off, and in few countries fourth wave is already coming, like in China or Korea. So what is your opinion regarding this impending fourth wave in our country?
COVID has been very unpredictable, and most of the statements of predictions have been rendered incorrect by this virus. So it is very difficult to predict anything, but yes, we are now past the third wave, that is for sure. Our daily number of new cases has gone down to low double digits, 20, 30 cases, 40, 50 cases per day. That is the number for UP, daily number. But as we see across the map, we see that the numbers are rising very fast. In South Korea, we had huge number of cases, though the population of South Korea is 1/4 of the population of UP. The number of daily cases at one point of time was between 5 to 7 lakhs new cases per day. Same for most of the European countries. Germany is giving around 2.5 to 3 lakhs new cases per day. Then same for France, and UK are also witnessing some surge. And Hong Kong, there the system was very much compromised, very much overwhelmed, and admissions, oxygen supply, everything was compromised. And that was very recent, in very recent past, maybe a week or 2 weeks back. So now that the numbers are rising everywhere across the globe, though we cannot be sure whether there will be a fourth wave or when it will be, but we are keeping ourselves ready. We are preparing for any a rise in number of cases. In fact, as we are talking to you, as we are discussing this, our teams are moving to the field for having a full rehearsal of COVID facility preparedness on 28th and 29th of this month. So we are keeping ourselves ready and keeping our fingers crossed that the numbers, if there is any surge, should not be very high, and the situation should be manageable. The hospitalization should not be very much, and we should be able to manage the patients well. There should not be any unnecessary mortalities or any crisis sort of situation that we faced during the second wave. That is our focus. As far as the fourth wave is concerned, nobody can say anything when it will come, what will be the magnitude. That's my answer.
Thank you, sir. But you must have strategized something or thought of something, because the number currently we don't have like much cases, but we are continuing to screening those patients, like 2,000, 4,000. So what is your strategy? How long we will continue to do that if the number doesn't increase at all? If numbers remain same or decrease, it's over now.
at least for next 12 weeks, 3 months or so. We will continue with the same number of tests. We will continue with the same level of preparedness. And if there is no further increase after 3 months or so, then we will think of relaxing a bit.
Then may I have one request?
Yes, please.
That you provide the same number of the staff, because the staff is going to stop from this 1st
I
April.
know. We are going to discuss this thing first thing on Monday morning. We are going to discuss the HR situation, because 31st of March your contract with your staff will end.
Yeah.
So
Yes, sir.
I know.
So everybody will go. Then how do we test? So. So thank you all. I think we had learned a lot from you and had a fruitful discussion. So I would like to thank all the panelists, Dr. Rika Sindhu, Dr. Singh, Vajanti Mala, Dr. Atul. Thank you all.
Thank you, ma'am. Thank you.
Thank you.
Thank you.
Thank you, sir. Thank you, sir.
Thank you.
So all good things come to an end. So this was the last session. So we started at around 10. Now it is more than 3, but I think it was like these 5 hours we spent wisely, and we learned a lot and enjoyed. We enjoyed in arranging. Hope our delegates also enjoyed it. And as I told you, we had more than 1,000 participants. Not only from various parts of India, northeast, east, west, south. Apart from this, surprisingly, we had 3 from Russia, around 8, 10 from US, and 1 from even West Indies. So I would like to take this opportunity to thank them all. They took interest, they took time out of their busy schedule and came here. Of course, I would like to thank American Society of Microbiology for giving me this opportunity, and Indian Virology Society for joining hands, because this is what COVID had taught us, that if we join hands, we can do anything. Then also Gadwasu University and my PhD student, Dr. Prabhat Ranjan, who's now in Alabama. I don't know, it's quite late, maybe he has slept, but I would fail my duty if I don't thank him. He helped me a lot. Then IDSP, ICMR, Government of India and UP, those who provide all the support in terms of manpower, infrastructure and everything. So, and they are doing, and so far we could do 3 lakh tests because of their big help. Then we have a number of publications also. So I would also like to thank our collaborators, especially from we have collaborators from CCM, from gastromedicine, you all know, then CDRI and others. So I would also thank. Then I should thank our own people. So co-in-charge Dr. Atul Garg, other faculty members, Dr. Nidhi, Dr. Dicha, Dr. Mugunthan, then the other Chinmoy, Sagram, they all help. They are all helpful and help me in every stage. But I will fail in my duty if I don't acknowledge the support of Mr. Nikhil Sharma, who's here, Mr. Nikhil, Miss Ankita, and of course, Nikki and all polio staff, then all the SRs, especially Dr. Akshay, who helped me in preparing slides, Dr. Radha is sitting there since morning, and other SR, JRs, technical staff, NHM staff who are working so hard for past 1.5 years or close to 2 years. Then polio staff, staff of Department of Microbiology, everybody. And my apologies if I forgot. And of course, telemedicine people, they're sitting there since morning. I must thank him and Mr. Rapudaman, whole telemedicine staff, because this was obviously not possible because of their active support and help. So I would thank each one of you. Thank you. And my apologies if I forgot to mention anybody. Buddha, Tanisha, they all are very supportive, and they are, that they are sitting since the morning. So thank you all. And this is what COVID has taught us.
Thank you.
So
one big clap for Dr. Ujala.
Sir, Ujjwala. And if not Ujjwala, actually it's a Bangla word. In Hindi, its equivalent is Ujjwala. So and of course, Dr. Yashpal Malik joined hands from the beginning and supported, gave ideas from the beginning at every stage. As he told, we are in close contact for past 2 months. So thanks to IBS, I told Dr. Yashpal Malik, Gadwasu.
Thank you very much, and each and everyone associated with this program.
Thank you, Dr. Malik. Thank you.